So this is chapter 24, the digestive system. And we'll start with an introduction and an overview and talk about the functions. So I'm not going to read everything on this slide. It is in the PowerPoint, so you can go back and look at it later in more detail. But basically, this is a long chapter with lots of material and lots of organs. So do not procrastinate studying this material and try to find ways to help yourself organize the information so it doesn't feel so overwhelming. So this is in the PowerPoint for you to read at your leisure. So in order for our mitochondria to make all of the necessary ATP to power all of our chemical reactions and functions, it needs oxygen, as we talked about in chapter 23. So the reason we breathe is to bring in the oxygen and get rid of the carbon dioxide. But there is another important component that is also needed to make ATP, and that is nutrients like glucose. And so the focus of this chapter is the digestive system and how we get that chemical fuel for all of the reactions in our body. So our digestive system allows us to get the nutrients such as carbohydrates, fats, and proteins that are needed to create the ATP we need, and also to provide building blocks for cell growth and repair. So keep the overall goal in mind as we go through this chapter. Even though there are a lot of organs and a lot of parts of the digestive system, the overall goal is to break down large molecules into their subunits so they can be absorbed into the body. So for example, if you eat sugars, they're broken down. Even a sucrose, which is a disaccharide, has to be broken down into monosaccharides. Lactose from milk has to be broken down into monosaccharides. Starch from all of your starchy foods have to ultimately be broken down into glucose. And then things like proteins have to be broken down into amino acids. Fats have to be broken down into triglycerides, etc. So we can't absorb the food as we eat it. So a big part of the digestive system is getting the food into a form that we can absorb into our bodies. So the digestive system has two main classifications of parts that belong to it. The first is the digestive tract, which is a long muscular tube. It's also called the gastrointestinal or GI tract or the alimentary canal. And so all of the organs that are actually part of this one long single tube is going to include the oral cavity, the pharynx, the esophagus, the stomach, the small intestines, and the large intestines. So even though they all are part of one long tube, we are going to talk about them separately. And then we have accessory organs, which are organs that assist with digestion in various ways. And these include the teeth, the tongue, the salivary glands, the liver, the gallbladder, and the pancreas. And again, we are going to be talking about each of these separately. So that's why I said lots of organs in this chapter. So the overall main function of the digestive system is to break down food, absorb the nutrients, and eliminate the remaining waste. So this primary function can be broken down into 6 integrated steps. Number 1 is ingestion. So this is when the food enters the oral cavity. So when you put food or drink in your mouth, this is an active process, so you have a conscious choice over what you are going to eat or drink. The second integrated step is mechanical digestion. This is the crushing and shearing of the food molecules to increase surface area. Things that you drink don't typically require mechanical digestion, but I will also point out that there are several organs along the digestive tract that participate in mechanical digestion. So as we move through this chapter and look at all of these different organs, one way you could help to organize your study is to come back to these 6 integrated steps. And as we look at each organ, ask yourself, okay, what is this organ doing as part of the overall big picture? So the third step is chemical digestion, which is breaking the chemical bonds in large molecules. So for example, when we eat something that contains protein, that protein molecule, which is a chain of amino acids, actually has to have chemical bonds broken so that we can get individual amino acids, which is the form that we absorb into our bloodstream. We then have secretion, which is the release of water, acids, enzymes, buffers, and salts, and this happens all along the digestive tract, as we'll see. We also have absorption, which is movement of those small organic molecules like amino acids across the digestive epithelium and into the body's fluids. And then we have defecation, which is removal of the waste products from the body. So after we have extracted everything we can use from what we eat, what is left over must be excreted. So there are also some protective functions of the digestive tract lining. So the lining of the digestive tract is also going to protect against the corrosive effects of acids and enzymes. So if you think about it, we actually have acids and enzymes to break down all of the food we eat, but we don't want to break down our own body's organs that are containing these acids and enzymes, so we have to protect ourselves against them. We also have protection against those mechanical stresses and abrasion that happen during the second step of mechanical digestion. And we also have to protect against bacteria. So there is bacteria that lives in our digestive tract, and there is bacteria that could be swallowed in the food or drink that you are consuming. In this section, we'll look at the peritoneal cavity and the mesenteries. So the peritoneal cavity is part of the abdominopelvic cavity, and it is lined by the peritoneum. So the peritoneum is another one of these serous membranes, like the pericardial membrane and the pleural membranes that we have talked about before. So there is a visceral peritoneum, which is the layer of the peritoneum that covers the organs in the peritoneal cavity. And so in this picture, for example, if this was one of the organs inside the peritoneal cavity, the visceral peritoneum is going to be attached to the outside of that organ. The parietal peritoneum is going to line the inner body wall surface. And then if you're interested in knowing which organs are covered by visceral peritoneum, so organs that are completely covered by the visceral peritoneum includes the stomach, most of the small intestines, the jejunum, ileum, and the first part of the duodenum. Also part of the large intestines, so the transverse colon and sigmoid colon, the liver, and the spleen. And then, as we'll mention in a few minutes, some of the organs of the digestive tract sit behind the peritoneal cavity, so they sit behind the parietal peritoneum, and they are called retroperitoneal. And that they are actually going to include a portion of the duodenum, the pancreas, and then these are some other organs like the adrenal glands, the ureters, the kidneys, and the rectum that are also retroperitoneal. So between the two layers of membrane, so between the parietal peritoneum and the visceral peritoneum, is where you would find the peritoneal fluid. And the purpose of this fluid is to reduce friction from all of these organs rubbing up against each other. And you actually secrete and reabsorb 7 liters of peritoneal fluid every day. So there's quite a lot of fluid that is surrounding your peritoneal organs. Ascites is a condition in which you get a buildup of peritoneal fluid that causes the abdomen to swell outwards. This can be caused by liver disease, kidney disease, heart failure, or starvation. And we talked before about why it can happen with heart failure. So because we've got multiple organs within the peritoneal cavity, and we've got the visceral peritoneum around the organs and the parietal peritoneum actually lining the cavity, all of these membranes are actually continuous. So there has to be a connection between the parietal and visceral peritoneum, and this double sheet connection is called the mesentery. So the mesenteries are these double sheets of peritoneal membrane that connect the parietal and visceral peritoneum. So here's an image from your textbook that shows the development, so the very initial development of the digestive system. So you can see that as it develops, you have the parietal peritoneum that is lining this cavity, and then you develop the visceral peritoneum that is around the organs, but then there has to be connections between them. And the connections that are joining the visceral peritoneum with the parietal peritoneum, again, these are double membrane connections called mesenteries. So the mesenteries provide a route for blood vessels, nerves, and lymphatic vessels to get to and from the organs. So if you think of the organs as kind of being suspended in this cavity or space, you have to have a place for blood vessels, nerves, and lymph vessels to get to and from them. The mesenteries also help to stabilize the position of the organs that are attached to them. And they also prevent the entangling of the intestines during digestive movements or changes in body positions. So a little later on, we're going to see just how long your small intestines are, and so you don't want them to tangle up and get knotted. So when you're doing your dissection, so you did the rat dissection in A&P1, you're doing the fetal pig dissection in A&P2, you can see the mesentery, so you can see this very thin membrane that is joining the organ to the wall of the peritoneal cavity, and you can actually see the blood vessels running through it to get to and from the organs. So the mesenteries can be divided into different sections depending on which organs they are connected to. So if we start up here at the very top, the falciform ligament is connecting the membranes of the liver to the parietal peritoneum, and the falciform ligament is helping to stabilize the position of the liver. Then we have the lesser omentum shown here, which runs between the liver and the stomach, and the lesser omentum helps to stabilize the stomach. The greater omentum runs from the stomach to the transverse colon, which is part of the large intestine, and it forms like this pouch or apron that drops down in front here. And you can get a buildup of adipose tissue in the greater omentum, and adipose tissue in this area contributes to the appearance of a beer belly. So you can see here, it's also called visceral fat. So the fat that is building up in that greater omentum is called visceral fat. So that's different than subcutaneous fat, which is fat that is just under your skin layer. So the purpose of having this fat is to provide insulation and as an energy reserve. But of course, everything in moderation, you don't want too much, because then that also causes its own problems. The mesentery proper is going to be all of the little sheets of mesentery that suspend the small intestine and helps to prevent it from tangling up. So when you're looking at the mesentery in lab in your dissections, you're actually looking at the mesentery proper. And then the last type of mesentery is the mesocolon, and there's some shown here. The mesocolon is going to be associated with the large intestine. A couple of other definitions. So again, when we say that something is retroperitoneal, we are referring to organs that sit behind the peritoneal cavity and behind the parietal peritoneum. And examples that we'll be talking about in this chapter are the duodenum, which is the first part of the small intestines, and the pancreas. Peritonitis is when you have inflammation of the peritoneum, and this can be a very dangerous condition and can even be fatal. This is why you don't want to leave appendicitis untreated, because if the appendix ruptures, that can cause peritonitis. Peritonitis can also be a complication of any surgery that goes into the peritoneal cavity, and then any damage that perforates the stomach or intestines can also lead to peritonitis. In this section, we will look at the layers of the digestive tract wall. So there are 4 layers altogether, and then we'll go through them one by one. But if we look at these 2 pictures and we start on the inside, so the innermost layer is called the mucosa, which is shown here, and then this would be the mucosa down here. Then underneath the mucosa, you have the submucosa, again shown in both pictures. Then you have a muscular layer. And then you have your outermost layer, which is called the serosa. And this is actually the same thing as the visceral peritoneum. So the exact structure of these 4 layers do have some regional differences. So for example, the muscular layer normally only has 2 layers of muscle, but when we take a look at the stomach, you'll see the stomach has an extra muscle layer. The small intestines has these folds called circular folds. They also have these even smaller folds called villi that are shown here in this bottom image. The circular folds can also be called plicae circularis. And the function of both the circular fold and the villi are to increase the surface area available for absorption. And so again, that's something that you'll see in the small intestines, but not necessarily in the other areas. And then secretions by glands that are either in the mucosa or the submucosa are carried up to the epithelial surface lining through ducts. So these are going to be mostly exocrine glands. And remember, the difference between an exocrine gland and an endocrine gland is endocrine glands secrete hormones into the bloodstream, and exocrine glands secrete hormones onto an epithelial layer. All right, so let's look at these 4 layers in a little more detail. And I've taken the picture from the textbook and I have grayed out the other layers so that we can just focus on the one layer we're talking about. So we're going to start with the mucosa. And this is a mucous membrane, just like we saw in the respiratory tract. So as a mucous membrane, it has an epithelium, and underneath it, it has a lamina propria. And then what makes this mucus layer a little bit different than the one that you see in the respiratory system is that you also have a thin layer of muscle underneath the lamina propria. So the epithelium in the digestive tract can be simple or stratified depending on the location. So in places where you have a lot of abrasion, like the oral cavity, pharynx, esophagus, and anus, you would have a stratified squamous epithelium. And then in areas that aren't subjected to a lot of mechanical stresses, like the stomach and the intestines, you would have a simple columnar epithelium. The epithelial cells of the digestive tract do take a beating, so with all the mechanical processing and chemical digestion going on, the epithelial cells lining the digestive tract are constantly having to be regenerated and replaced. This is one of the reasons why chemotherapy treatments for cancer, which stop the ability of all cells to divide, drastically affect the digestive tract, and this is why those drugs are usually, their big side effects include nausea and vomiting. So in the epithelium, you can also find mucus cells in some areas and goblet cells in others, and both of these cell types secrete mucus. You can also find the odd enteroendocrine cell, which is a cell that is embedded in the epithelium that secretes hormones that can regulate the digestive system activity. And then underneath that epithelial layer, we have the lamina propria, which is areolar tissue that contains blood vessels, lymphatic vessels, and nerves. And you may find the occasional gland in this area as well. And in this area, you would find the glands called intestinal glands or intestinal crypts. And then underneath the lamina propria, you have this thin, smooth muscle sheet called the muscularis mucosae. So notice the name mucosae is telling you that it is in the mucosa. And in this muscle layer, we have both circular muscle and longitudinal muscle. And this muscle functions to move the folds, like these circular folds within the small intestine. And it can also alter the shape of the lumen. So make the lumen more narrow or cause it to have ridges in it. So the next layer is the submucosa, and again on the picture I have highlighted it for you and grayed out the other areas. So the submucosa sits underneath the mucosa, and it is made up of dense irregular connective tissue. Here is where you would find numerous blood and lymphatic vessels. Some regions of the digestive tract will also have some exocrine glands located in this area, like here's a submucosal gland in this particular image, and you can see how its little duct would cause the secretions to go out to the epithelial surface. And there is a network of nerves in this region shown by the yellow lines called the submucosal neuroplexus. And this is part of the enteric nervous system, which I'll explain a little bit more about on the next slide. But when you're remembering where the two different layers of nerves are located, the name of the nerve plexus gives you a clue. So the submucosal neuroplexus is located in the submucosa. So the next layer is the muscular layer, and this is made up of a bunch of smooth muscle cells. And the muscle in this area is also oriented in two directions. There is circular muscle and longitudinal muscle. So if we take a look at the two different types of muscle, so circular muscle is going to wrap around the digestive tract, so in a circular motion. So when it contracts, it will squeeze and close the lumen or the passageway in the middle of the tract. The longitudinal muscle is going to run lengthways along the tract. So when it constricts, it will shorten the portion of the digestive tract where it is constricting. In between these two muscle layers is another layer of nerves called the myenteric plexus, which is also part of the enteric nervous system. And if you remember that the prefix myo means muscle, that will help you remember that the myenteric plexus is in the muscular layer. And then the outermost layer is called the serosa, and this is also known as the visceral peritoneum, which is the part of the peritoneal membranes that lines the organs. So just a little bit more about the enteric nervous system, or ENS. This is a division of the autonomic nervous system, and it can get quite complex, and don't panic at looking at this particular image. I just want you to notice that you have the two levels of nerves. So you have a submucosal plexus that's in the submucosa, and the myenteric plexus, which is in that muscular layer. And you can see the myenteric plexus is buried between the longitudinal muscle and the circular muscle. The submucosal plexus is going to sit underneath that muscularis mucosae, which is part of the mucosa layer. And so these neurons are connected to each other. They're connected to sensory systems, endocrine cells, a bunch of different types of receptors, and sympathetic and parasympathetic pathways. So the enteric nervous system is actually turning out to be the largest and most complex part of the peripheral nervous system. So we just get a little paragraph about it in our textbooks now, but I can guarantee you in the future, as we learn more and more, you're going to have a whole chapter just on the enteric nervous system, just like you have a whole chapter on the autonomic nervous system. So outside of the brain, it has the largest number of neurons. So some estimates have said 100 million, but they have since raised that to 600 million. So 600 million neurons and many different neurotransmitters, including serotonin, the same neurotransmitter that plays a role in depression. And now there seems to be a close link between depression, anxiety, and irritable bowel syndrome because of the interaction between the brain and the enteric nervous system. I'll put an article on D2L if you're interested in learning more about the ENS. So the ENS is getting input from sensory neurons, chemoreceptors, and mechanoreceptors that can sense what is going on inside the digestive tract. And you've got parasympathetic and sympathetic connections. And just as a reminder, the parasympathetic nervous system promotes digestion and motility. That is why it's called the rest and digest division. And the sympathetic nervous system decreases digestion and motility because this is your fight or flight response. And while you're running away from a lion, you don't want to have to worry about digesting your food. Now we'll look at the motility of the digestive tract, and the two main types of motions are peristalsis and segmentation. So smooth muscle cells have the ability to self-contract just like the cardiac cells we talked about earlier in the semester, and this is a property of automaticity. So in the digestive system, there are pace setter cells which can undergo spontaneous depolarization and fire an action potential, which will start a wave of contraction that can move through the smooth muscle in both the muscularis mucosae and the muscular layer. So the pace setter cells enable rhythmic cycles of activity, and again, they can be found in both the muscularis mucosae and the muscular layer of the digestive tract. So the first type of movement that is produced is called peristalsis. This is when waves of smooth muscle contraction propels materials along the lumen of a tube. And we're going to see peristalsis in some other systems as well, like the urinary system and the reproductive system. But this is the first place that we're seeing it, so we'll go into a little bit more detail. But keep in mind, peristalsis is moving things, so it's pushing it forward. And then with reverse peristalsis, it can actually move it back the other way. So waves of contractions in the digestive system move a bolus along the digestive tract. And a bolus is just a soft, rounded ball of digestive contents. So after you swallow a mouthful of food, that then becomes a bolus. So as the bolus is moving from the mouth all the way to the anus through the digestive tract, remember we've got these two layers of muscle, longitudinal muscle and circular muscle. And so the circular muscle is contracting behind the bolus, and the longitudinal muscle is contracting ahead of the bolus. And so with the one pushing from behind and the other pulling from the front, this moves the bolus forward through the digestive tract. So even if you were to stand on your head and so you didn't have gravity helping you, food would still be able to go down your esophagus, for example, and move through the rest of your digestive tract. And that is because of the smooth muscle contractions causing peristalsis. There is also a movement called segmentation, which is contraction of digestive smooth muscle with no set pattern. So again, this is cycles of contraction with no set pattern. So the purpose is not to move anything, but rather to keep the food in the same place and mix it up. So this is a type of churning, which plays a role in the mechanical digestion process. It can break the bolus into smaller pieces, as shown here in A and then B. And then it can also mix the contents of the bolus with the intestinal secretions, so with things like buffers and enzymes. And so you get this mix when you're all done of very small particles that are all mixed in with the enzymes needed to chemically digest them. So as a reminder, peristalsis is all about moving stuff forward, so propelling stuff in one direction, whereas segmentation kind of keeps it in the same direction and just mixes and churns it. So we're going to go ahead and introduce the regulation of the digestive system. Regulation is usually covered at the end of chapters, but because we have so many organs and structures in the digestive system, we're going to talk about a lot of this as we go through the different organs and structures, so we're just going to introduce it at this point. And there are 3 levels of digestive system regulation. So the first level is going to be local regulation, and this is when the pH, volume, or chemical composition of the contents inside the intestines can change the activity of that area of the intestines. So for example, stretching of the intestinal wall, which can be sensed by baroreceptors, can stimulate local muscle contractions that might allow segmentation or peristalsis in that area. And we're going to see how this works as we go through the digestive tract, again, as we get to the different organs and parts of the digestive tract. Then there is also neural regulation. So we've already briefly talked about the enteric nervous system, which is a division of the autonomic nervous system, and the nerves that are located in 2 areas, the myenteric plexus in the muscular layer and the submucosal plexus in the submucosa. And then there are also neural reflexes. So short reflexes are reflexes where the action potentials don't ever leave the enteric nervous system, so they stay entirely localized. So for example, secretions of digestive glands and local peristalsis could be triggered in response to the arrival of a bolus to a particular area. Long reflexes are going to actually send signals to the central nervous system, so to the brain or the spinal cord. And so, for example, larger scale peristalsis, like moving materials from the small intestine to the large intestines, would be a reflex that is controlled at a higher level. And then there is also hormonal regulation. And so, as usual with any type of hormone, these are secretions into the bloodstream. And in this case, these secretions would then regulate digestive system functions. And we're going to talk about the hormones as we proceed and we get to specific organs. And then we're going to review them all again at the end. So again, this is just an introduction to the 3 different levels of digestive function control. And we'll talk more about these different control mechanisms in the subsequent sections. So there are local factors. So these are things like the actual pH changes that take place inside the lumen. Then you have your neural control mechanisms. So you have your 2 different branches of the enteric nervous system. So the submucosal plexus and the myenteric plexus. You've also got your short reflexes, which stay local and never actually leave the enteric nervous system, and then your long reflexes, which go up to the central nervous system. And then with hormonal control mechanisms, we have things like the enteroendocrine cells that release hormones that go into the bloodstream, which then come back and change the activity of the digestive system. In this section, we'll be looking at part 1 of the oral cavity, which will include the teeth and the tongue. And I'm breaking this into multiple sections because I don't like any individual section to get too long. So we'll start with the oral cavity. And the oral cavity is also known as the mouth, and it has several functions. So it provides sensory analysis of food before you swallow it. It also assists with mechanical digestion by actions of the teeth, tongue, and hard palate. It helps with lubrication of what you're chewing by mixing the food items with mucus and saliva. And it allows for limited chemical digestion. So the initial digestion of carbohydrates and lipids starts in the oral cavity. So the oral mucosa, which is the mucosa portion of the mucous membrane that lines the oral cavity, is a stratified squamous epithelium. No absorption takes place in the oral cavity except for a small area under the tongue where the mucosa is thin enough and vascular enough to absorb lipid-soluble drugs only. So this is why there are some medications you put under your tongue to dissolve, but the majority of absorption is going to happen later on in the digestive tract. So let's look at some of the structures in the oral cavity. I'm not covering every structure that's listed in your textbook because this chapter is already long enough. But the first structure we'll look at is called the gingivae, or this is also known as your gums. These are the ridges of oral mucosa that you find around the base of each tooth. The hard palate we've mentioned before. This is made up of the maxilla and palatine bones, so it's the bony roof of the mouth. And the hard palate has ridges that can actually provide traction, which helps with the mechanical processing of food as you're chewing it. The soft palate is the soft portion behind the hard palate where there is no bone. It's also the border between the nasopharynx and the oropharynx. And the little dangly part that comes off the back of the soft palate that you can see dangling at the back of your throat is called the uvula. So this is an extension of the soft palate that helps prevent food from entering the pharynx too soon. The fauces is shown here. This is the border between the oral cavity and the oropharynx, which is right behind the oral cavity. The frenulum is the term for the little folds of mucosa that extend from the gingiva to the upper and lower lips. So there's a frenulum of the upper lip and a frenulum of the lower lip. You can usually fill these with your tongue. So now let's look closer at the tongue. So the tongue has some functions of its own. It assists with mechanical digestion by compressing, abrading, and distorting the food as you move it around in your oral cavity. It also helps with a manipulation to assist in chewing and to prepare food for swallowing. It also provides sensory analysis for touch, temperature, and taste through the gustatory receptors that we covered at the beginning of the semester. And it secretes mucins and lingual lipase. So the lingual frenulum, or frenulum of the tongue, is that little bit of tissue at the bottom of your tongue. It's a thin fold of mucous membrane that connects the body of the tongue to the mucosa on the floor of the oral cavity. Ankyloglossia is when you are born with a shortened lingual frenulum that causes the tongue to be partially or completely fused to the floor of the mouth. And this usually has to be surgically corrected because it can interfere with your ability to eat and speak. And this is actually where our saying to be tongue-tied comes from, because people who are born with this mutation that caused their tongue to literally be stuck to the bottom of their mouth, they were called tongue-tied. So lingual lipase, as I mentioned, is one of the secretions of the tongue, and this is an enzyme that is going to begin the digestion of lipids. So lipase is an enzyme that breaks down lipids, and lingual tells you that it's made by the tongue. So lingual lipase. And then the mucins that are also secreted by the tongue are glycoproteins that function as lubricants. So it mixes with the food and then makes it slide easier down your throat and through the digestive tract. Now let's look at the teeth. So we're going to start with the anatomy of a tooth, and your teeth are used for chewing food. So the bulk of the tooth is made of dentin, so that's this part here. Dentin is a mineralized matrix that is very similar to bone that we covered in A and P1, except that it doesn't have any cells, so no osteocytes, and it has little canals that run through it that connect the dentin to the pulp cavity. And the pulp cavity contains blood vessels and nerves that enter at the bottom through the small opening called the root canal, and the part of the tooth that is buried in the bone is called the root. And the teeth sit within a bony socket in the bone, so the maxilla for the upper teeth, the mandible for the lower teeth, and this bony socket is called the tooth socket or the tooth alveolus. So the tooth is connected to the bone by the periodontal ligament. So this ligament connects the teeth to the bone, and this type of joint is called a gomphosis. That was also covered in A and P1. And then a thin layer of cement covers the dentin on the root to anchor it to the periodontal ligament. The crown is the exposed portion of a tooth, so the portion that you can see. And the dentin in the crown is covered by a substance called enamel. And enamel is made up of crystallized calcium phosphate, and it's the hardest biologically produced substance. So it's the hardest substance that is actually produced by a biological organism. The gingival sulcus is going to be this shallow groove here that surrounds the neck of each tooth. And at this point, the mucosa epithelium is actually directly bound to the tooth at the base of the sulcus. And this prevents bacteria from having access to the lamina propria, which is the layer of connective tissue, areolar connective tissue of the mucous membrane. When you brush your teeth, you help to strengthen this attachment, which makes it less likely that bacteria can break through. Gingivitis is when you have inflammation of the gingivae, and this is caused by a bacterial infection from where bacteria are able to get past the barrier and into the tissue that makes up the oral mucosa. So bacteria can adhere to the surface of our teeth, and they produce a biofilm, which is called a dental plaque. And then once they have that biofilm in place, they secrete acids underneath it, and those acids can eat away and dissolve the tooth enamel, even though it's an extremely hard substance. And then the dental plaque, which is soft to begin with, can become calcified, and then it's forming a hard layer of what is called tartar or dental calculus. And once it becomes calcified and hardened, it becomes much harder to remove. Tartar deposits most commonly develop near the gingival sulcus, which is the groove around the neck of the tooth, where brushing cannot easily remove the soft plaque deposits, so they stay there until they have a chance to harden. So what happens during tooth decay is you start with these plaque buildups that become tartar over time, and then the bacteria are able to eat away at the enamel and cause caries and deep cavities. And so in the initial stages, when the cavity and decay is just affecting the enamel, you don't feel any pain, but you still should have your teeth checked, because these are the stages at which it is best to have these holes filled and prevent further bacterial decay, because once the bacterial decay reaches the dentin and then the pulp, that is when things become very painful. So now let's look at the type of teeth, but first let's look at a couple more definitions. So the occlusal surface is the opposing surfaces of the teeth that come into contact when chewing. So like if you were to run your finger over the bottom of your teeth, you are feeling the occlusal surfaces. And the cusp are these little elevations or projections of the occlusal surface. So now if we look at the four types of teeth, the first type are the incisors, shown here on the left. These are blade shaped and they have a single root, and these are used for clipping or cutting. So like when you're biting into an apple or a corn on the cob, for example. The next type of tooth is a canine, which is also called a cuspid. This is a conical shaped tooth with a single pointy cusp. And in animals like your cats and dogs, this tooth can get quite long, and so that's why we call those long pointy teeth at the front of their mouths canines. And they have a single root as well, and these are used for tearing or slashing. The next type of teeth are the premolars, which are also called bicuspids. These have a flattened crown with two cusps, which you can more easily see in this picture here. You can see the two cusps, one on each side, and they have one or two roots, and these are used for crushing, mashing, and grinding. And then you have your molars, which are the ones in the back of your mouth, and they have very large flattened crowns with 4 to 5 cusps and 2 to 3 roots, and they are used for crushing and grinding. So teeth are collectively called the dentition, and your first dentition are called the deciduous teeth. Sometimes they are also called the primary teeth, the milk teeth, or your baby teeth, and the deciduous teeth are shown up here on the left side. So you have 20 total teeth, and when we are counting the teeth and looking at the different types you have in each type of dentition, it helps to break the teeth up into quarters. So if I draw a little box so that I'm only looking at 1/2 of one side, and then I can see easily that in my primary dentition or my deciduous teeth, I have 2 incisors, 1 canine, and then a pair of molars that are called deciduous molars. Then you have your secondary dentition, which are called your permanent teeth. Eruption is the emergence of the permanent teeth, which pushes out the deciduous teeth. Your permanent teeth, you have a total of 32 of them. And again, if we look at the different types, it can help to draw a square and just look at a fourth. So your permanent teeth, you have 2 incisors, 1 canine, 2 premolars, and then 3 molars. And so notice that since I'm looking at a fourth, this is a total of 8 teeth. 8 times 4 is 32. Just like up here, this was 5 teeth. 5 times 4 is 20. So that third set of molars are called the wisdom teeth, and they are the last to erupt. They typically don't erupt until you're between 17 and 21 years old. And in some people, they never erupt because there's not enough room for them to come out, and you may have to have them surgically removed. And so an impacted tooth is any tooth that develops in a location where it cannot erupt, and that does happen to the wisdom teeth in some people. So now we'll do the oral cavity part 2. We'll look at the salivary glands and then at mechanical digestion, mastication. So the salivary glands are glands that secrete saliva, and you have 3 major salivary glands. The first ones are the parotid glands that sit off on the side of your face. This is the area where you feel tingling if you put something sour in your mouth and you get that tingly feeling right in front of your ear. That is actually your parotid glands. The parotid glands secrete large amounts of salivary amylase, and it drains into the oral cavity through the parotid duct, shown here. Then you have the sublingual glands, which are underneath the tongue. And so sub means under, lingual refers to the tongue, so sublingual is under the tongue. These glands secrete buffers and lubricants. And then you have the submandibular glands, which are on the inner surface of the mandible, so think under the mandible. And these glands secrete buffers, mucins, and some more salivary amylase. There are also 800 to 1,000 minor salivary glands, which are part of those submucosal glands. So they're in the submucosal layer of the oral mucosa. So salivary amylase is an enzyme that begins the digestion of starch, which is a carbohydrate. Mumps is a viral infection of the salivary glands. In males, mumps can spread to the testes and cause sterility. Serious cases of the mumps can also spread to other systems like the central nervous system. And because it is a dangerous disorder, there is a vaccine that you can get, and it's normally given to children. So let's talk about saliva. So you believe it or not, you actually make 1 to 1.5 liters per day. So imagine a 1 liter Coke bottle, and you make 1.5 of those per day of just saliva. 70% of your saliva comes from your submandibular glands, 25% comes from the parotid glands, and the remaining 5% comes from the sublingual glands. Saliva is mostly water, so it's 99.4% water. The other 0.6% is a combination of electrolytes, including sodium ions, chloride ions, and bicarbonate ions. You also have buffers, mucins, antibodies, enzymes, and waste, all in that 0.6%. The buffers help to keep the mouth pH around 7, which is a neutral pH, and this helps fight against the acidic environment that is preferred by the bacteria that cause cavities in your teeth. Saliva also includes antibodies, and they are the type of IgA antibodies, which are the types of antibodies included in bodily secretions. And these antibodies also help to control oral bacteria. And saliva can also contain lysozymes, which we discussed back in chapter 17 when we talked about the composition of tears. And the lysozymes can also help control oral bacteria. In fact, if you have any kind of condition that reduces the production of saliva, so you make less saliva than normal, this can actually trigger an overgrowth of oral bacteria. So any object placed in the mouth can trigger a salivary reflex. and this is controlled by the salivatory nuclei in the medulla oblongata. And because the medulla oblongata also has connections to higher brain centers, just the fact that you think about food or a food that you're craving, or if you're hungry, all of those things can make you start to salivate as well. So saliva functions to lubricate the mouth, to moisten and lubricate materials in the mouth, to dissolve chemicals so that they can bind to the gustatory receptors on the taste buds, and to begin the chemical digestion of complex carbohydrates with that enzyme we talked about called salivary amylase. Saliva may also contain a little bit of lingual lipase, which is secreted by the tongue, and as a reminder, this helps with the chemical digestion of lipids. So one of the stages of mechanical digestion takes place in the oral cavity in the process of mastication, and mastication is the anatomical term for chewing. So the muscles of mastication, which we talked about in A&P1, and which include the temporalis and the masseter muscles, help to move your lower jaw up and down and side to side to move food across the occlusal surfaces of the teeth. Fine control of the movement of the food is done with the cheeks, the lips, and the tongue. And the functions of mastication are to break down the food into smaller pieces, especially the tough connective tissues that you find in meats and the tough plant fibers that you find in vegetables. Mastication also helps to saturate food with the salivary secretions and enzymes. And then after mastication, your tongue compresses the food into a moist, cohesive bolus that is then easy to push to the back of the pharynx and swallow. In this section, we're going to look at the pharynx and esophagus and also talk about deglutition. So the pharynx or throat is a common passageway for solid food, liquid, and air. And it has three sections: the nasopharynx, the oropharynx, and the laryngopharynx. And we covered the boundaries of these different sections in chapter 23. It is, the epithelium in the pharynx is a stratified squamous epithelium. And the lamina propria has mucus glands and mucosa-associated lymphoid tissue, which are the tonsils that we talked about in chapter 22. The pharynx also has muscles that help with swallowing, and swallowing is the act of pushing the bolus of food down into the esophagus. So the esophagus is a hollow muscular tube that carries ingested material to the stomach. It is also lined with stratified squamous epithelium, and it has extensive folds that allow it to expand when swallowing. So if you look at a transverse section through an empty esophagus, you can see all of the folds that it has within its mucosa and submucosa. The resting muscle tone keeps it closed until you swallow, so the esophagus is essentially closed when it is resting. The superior esophagus has skeletal muscle in addition to smooth muscle. And even though it doesn't have well-defined sphincters, we still use this terminology to describe the upper and lower areas of the esophagus where you have active contraction of the muscles. So the upper esophageal sphincter, or UES, is going to be at the top of the esophagus. And the lower esophageal sphincter, or the LES, is going to be at the bottom of the esophagus right before it empties into the stomach. And this, the older terminology for the lower esophageal sphincter was the cardiac sphincter, because the top part of the stomach is called the cardia. So if you see that in literature or other places, realize that that is referring to the lower esophageal sphincter. The esophageal hiatus is the opening in the diaphragm where the esophagus passes through. And there are esophageal glands along the esophagus that produce mucus to reduce friction and help the bolus of food to slide smoothly down the esophagus. So deglutition is the act of swallowing. It is both voluntary, meaning you can decide when you want to swallow, and it is a reflex. So once you trigger certain receptors in the back of the pharynx, it then the reflexive action takes over. So the swallowing reflex is triggered by tactile receptors in the oropharynx, and the swallowing center that controls the reflex is in the medulla oblongata. So as part of the swallowing reflex, you have inhibition of the respiratory centers, which stops your breathing because you're going to be closing the epiglottis over the opening to the trachea, so you won't be able to breathe at the same time that you swallow. You swallow approximately 2,400 times per day. So obviously you swallow when you're eating, but you also swallow in between meals. Anytime saliva builds up enough in the back of your throat to trigger the swallowing reflex, you swallow that saliva without even thinking about it. So the act of swallowing has 3 phases. The first phase is called the buccal phase. So buccal is another term for like the oral cavity. The buccal phase is the act of moving the food toward the back of the mouth. So the tongue is pushing the bolus up against the hard palate and then backwards into the oropharynx. And the soft palate is going to raise up to close off the nasal cavity. And once the bolus reaches the back of the pharynx, this is going to trigger that swallowing reflex. Then you have the pharyngeal phase. And during the pharyngeal phase, the larynx elevates, as we discussed in chapter 23, and when it elevates, the epiglottis closes over the glottis, which is the opening into the larynx. And then again, the uvula and the soft palate are blocking entry of the bolus into the nasopharynx or nasal cavity. Then during the esophageal phase, the bolus is in the esophagus and it is being pushed downward with peristalsis to the stomach. So then at the end of the esophageal phase, the bolus would then enter the stomach. So primary peristaltic waves are the normal peristalsis that pushes the bolus downward to the stomach. The normal trip for food from the oral cavity to the stomach takes about 9 seconds. Liquids tend to move faster. They can actually move ahead of the peristaltic contractions and are pulled downward by gravity. But if you poorly chew something or something you swallow is dry, it's poorly lubricated, it can travel down the esophagus much more slowly, and you actually have to have a secondary peristaltic wave, one or more secondary peristaltic waves, to push it into the stomach. And sometimes you can actually feel that. It feels like something is stuck in the esophagus in your chest, and you can feel the contractions of the muscles as it's trying to push it downward into your stomach. And the secondary peristaltic waves are triggered by local reflexes when sensory receptors in the esophageal walls sense that the food is still in there and it hasn't made it down with the initial primary peristaltic waves. So now we're moving on to the stomach. And in this section, we'll look at the stomach anatomy and its secretions and talk about stomach acid. So the stomach is an expandable tube-like organ in the upper left quadrant of the abdominal pelvic cavity, shown here in this image. The stomach has 4 main functions. It provides a place to temporarily store ingested food until the remainder of the digestive tract is ready for it. It also helps with mechanical digestion through muscular contractions that help to mix the food. It also assists with chemical digestion through the use of acids and enzymes. And it is the site of the production of intrinsic factor, which is required for vitamin B12 absorption. So after you have swallowed some food and it gets into your stomach and it is mixed with the secretions from the stomach, it is then called chyme. So chyme refers to the viscous, highly acidic, soupy mixture of partially digested food. The shape and size of the stomach can vary from person to person and even from meal to meal, depending on how much you eat. but typically the stomach has the shape of an expanded J. So if we look at the gross anatomy, the stomach has two main curves. So the greater curvature would be the largest curving side that is found on the lateral side of the stomach, and the lesser curvature would be the smaller curved side that is on the medial surface. And then we can divide the stomach into four regions. The first region is the cardia, which is up here at the base of the esophagus. This is why the lower esophageal sphincter used to be called the cardiac sphincter. And in the cardia, you have abundant mucus glands for protection from the acids and enzymes of the stomach, and that protection extends back up into the esophagus. And here's a picture to help you better visualize these three areas. So the cardia is the smallest of the four areas. Then you have the fundus, which is the area of the stomach that is superior to where it joins up with the esophagus. And the fundus region contains many gastric glands. And gastric glands are the ones that secrete the acids and enzymes, and we'll look in more detail in the gastric glands in a moment. The body is going to be the largest portion of the stomach, and this is the mixing tank where the food you eat is mixed up with all of these different secretions, and it contains gastric glands as well. And then the pylorus or pyloric part is this last part of the stomach right before it connects with the small intestine, and the first part of the small intestine is called the duodenum or the duodenum. So the pylorus contains pyloric glands, and we're going to look at these different glands in a moment. So at the end of the pyloric part, you have the pyloric sphincter, which is a strong region of circular muscle that can close off and regulates the release of the chyme from the stomach into the duodenum. So the stomach lining is full of a lot of folds called rugae, and these folds allow the stomach to expand as you eat, and so you can actually fit quite a lot of food into the stomach. The stomach can expand up to 50 times its size when it's at rest, and this is why you're able to go to all-you-can-eat restaurants, and why people are able to have like these hot dog eating contests and pie eating contests, because of the expandability of the stomach. Remember, one of the functions of the stomach was to temporarily store the food until the small intestines is ready for it. So when you eat a lot, it's going to sit in your stomach for a while until it gets emptied out into the small intestines. The stomach has an extra layer of muscle. So earlier we talked about the muscular layer and how it has a longitudinal muscle and a circular muscle layer, but the stomach has a third layer called the oblique muscle layer. And so this is an extra layer of muscle that is found in that muscular layer of the lining, and it's found only in the stomach. And it's going to help with the churning and mixing actions that the stomach does to mix the food that you eat with the acids and enzymes. So let's take a look at the stomach lining. So the lining in the stomach is going to be a simple columnar epithelium in the mucosa layer. And the stomach lining is covered by a carpet of alkaline mucus that will protect these cells from the acid and the enzymes that are going to be present inside the lumen or open portion of the stomach. So if we look closely at the mucosa of the stomach wall, there are these gastric pits up here. These are shallow depressions that open up onto the gastric surface. And the cells located in the gastric pits are constantly dividing to replace the epithelial cells that are lost into the lumen of the stomach. So being a stomach cell is a tough place to be. So gastric epithelial cells typically only live for 3 to 7 days. And it's probably not surprising that they have such a short lifespan, since the stomach is a pretty harsh environment full of acid and enzymes. Exposure to alcohol and certain other chemicals can actually increase the cell turnover and can actually make it more likely that you end up with an eroded mucosa, which can lead to what we call an ulcer. So gastritis is superficial inflammation of the gastric mucosa, and if gastritis persists for long enough, it can cause peptic ulcers, which is an erosion of the stomach or the duodenal lining from exposure to acids and enzymes. So a gastric ulcer is an ulcer in the stomach, a duodenal ulcer is an ulcer in the duodenum, and these are both types of peptic ulcers because they're caused by the peptic juices, and specifically the enzyme pepsin. So let's take a look at these gastric glands. So the gastric glands, as a reminder, are found in the fundus and the body of the stomach, and they are located at the bottom of the gastric pits. So here's the gastric pit here, and then at the bottom we can have one or more gastric glands. So gastric glands secrete quite a bit of fluid. They secrete 1.5 liters of gastric juice per day, and this is in addition to the 1.5 liters of saliva you are secreting per day. So there is a lot of secretion going on in the digestive system. So later we're going to talk about how important it is for us to get a lot of the water back that is a large component of these secretions. There are 2 primary types of secretory cells in the gastric glands. The first are the parietal cells. The parietal cells are the ones that secrete intrinsic factor, which is required for the absorption of vitamin B12. If you can't make intrinsic factor because of like a genetic mutation, you're at risk for pernicious anemia, which we talked about in the cardiovascular system chapters. The parietal cells also secrete hydrochloric acid, and hydrochloric acid is a very strong acid. So on the next slide, we'll go into a little bit more detail to see how the body can secrete this without actually harming itself. The purpose of the hydrochloric acid is to keep the stomach pH very low, so between 1.5 and 2, which is extremely acidic. The other major type of secretory cell is the chief cells. These guys secrete pepsinogen, which is an inactive proenzyme. So proenzymes are not yet quite all the way to the enzyme stage, so they are inactive and they require some kind of chemical modification before they can become an active enzyme. So when pepsinogen is exposed to an acidic environment, the acid converts the pepsinogen into pepsin, which is an active form of this enzyme. So peptin requires a pH of between 1.5 and 2 in order to become activated. And if you think about it, it makes sense for our cells to make an inactive form of an enzyme that breaks down proteins, because if you remember, most of our cells, most of the jobs inside of our cells are done by proteins. So if the chief cells tried to make an active form of pepsin, as soon as they made this enzyme, it would start breaking down everything inside the chief cell. So the chief cell secretes an inactive form, that way it doesn't harm the cell itself. And then once the pepsinogen gets into the acidic environment and the acid is secreted by the parietal cells, then that pepsinogen gets converted into pepsin, and pepsin chemically digests proteins by breaking the peptide bonds between the different amino acids. There is also cells called G cells that can be found inside gastric glands, but these are not secretory cells. These are enteroendocrine cells, meaning they are actually releasing hormones that will enter the bloodstream. And the G cells produce the hormone called gastrin, which promotes stomach activity. And we'll look a little bit more detail in this hormone in a few minutes. An interesting fact, I'm not going to test you over this, but you might be interested to know that babies, but not adults, also produce gastric lipase in their stomachs, and this is an enzyme that breaks down fats, and this helps babies to digest the fats found in the milk, since milk is the primary diet of a baby. All right, so let's talk a little bit more about the stomach acid. So, as mentioned before, the parietal cells are the cells in the gastric glands that produce hydrochloric acid. But hydrochloric acid is a very strong acid, and it is too strong to be made inside the cell. So if it was made inside the cell, it would actually destroy the cell. So hydrochloric acid is instead formed in the lumen of the stomach by an independent transport of hydrogen ions and chloride ions. So if we look first at the production of the hydrogen ions, so the hydrogen ions come from carbonic acid, and carbonic acid comes from the breakdown of carbon dioxide. So you remember that equation in chapter 23 that I said wasn't going away for the rest of the semester? So if you remember, carbon dioxide combines with water, and an enzyme called carbonic anhydrase converts the carbon dioxide and water into carbonic acid. Carbonic acid then immediately dissociates into hydrogen ions and bicarbonate ions. So this is where the hydrogen ions come from for the hydrochloric acid. The hydrogen ions are then actively transported into the lumen using active transport proteins. So it's going to take ATP because this is active transport. The chloride ions come from the chloride shift. So just like we saw with the red blood cells, after the carbonic acid dissociates into bicarbonate ions and hydrogen ions, the bicarbonate ions are exchanged out into the interstitial fluid for a chloride ion. So for every bicarbonate ion that leaves the parietal cell and goes into the interstitial fluid, a chloride ion comes into the parietal cell. So chloride ions come into the parietal cell through the chloride shift as they are exchanged for bicarbonate ions. The chloride ions then diffuse out the other side of the cell and into the lumen. And so now we have both the hydrogen ions and the chloride ions in the lumen to form hydrochloric acid. So the mix of hydrogen and chloride in the lumen keeps the pH of the stomach between 1.5 and 2, which again is very acidic. So the reason why we have stomach acid, so the functions of the stomach acid, is that it's going to kill most of the microorganisms ingested with food. And remember, our mucociliary elevator is also moving our respiratory mucus to be swallowed. And again, this is going to kill any microorganisms that were trapped in that mucus. It's also going to denature, which means to unfold proteins, and it'll inactivate most enzymes found in your food. So any enzymes in the food can't interfere with the digestive process. It's going to help break down the plant cell walls of vegetables, and it's going to help break down connective tissues in meats. And it is required for the activation of pepsin, which is that protein digesting enzyme that was secreted in its inactive form from the chief cells. So the proteins that are actively pumping the hydrogen ions into the lumen, this yellow circle on this picture, are called proton pumps, because hydrogen ions can also be called protons. And proton pump inhibitors are used to reduce the production of stomach acid. These are some of the most widely prescribed drugs these days. They're even available over the counter now. You may have heard of some of these like Prilosec or Prevacid. Prilosec is the brand name for omeprazole. So these are all very common anti-acid drugs that people are given to help control things such as acid reflux in the esophagus and heartburn. So we talked about the gastric glands. Now let's look at the pyloric glands. So the pyloric glands are located in the pyloric part of the stomach. The secretory cells in the pyloric glands primarily secrete mucus, but then the pyloric glands have a lot of enteroendocrine cells that produce several hormones, at least 7. We're only going to look at 3. So gastrin is produced by the G cells. G cells can be found in both gastric glands and pyloric glands. Gastrin stimulates the secretion of both the parietal and chief cells. Remember, parietal cells are making hydrochloric acid, chief cells are making pepsinogen. Gastrin also stimulates the muscular mixing actions in the gastric wall. And G cells are also located in the duodenum. So G cells all over the place that are secreting gastrin. Another type of enteroendocrine cell called a D cell secretes a hormone called somatostatin. Somatostatin inhibits the release of gastrin. So it's an anti-gastrin hormone. And somatostatin is normally continuously secreted unless it is stopped by neural or hormonal signals after you eat something and food enters your stomach. Then somatostatin is inhibited, and then that allows gastrin to be released. And then ghrelin is a hormone that stimulates the feelings of hunger. So it is a pro-hunger hormone, and it works opposite to leptin, which we talked about in chapter 18. So remember, leptin is a hormone produced by your adipose tissue, and it makes you feel full. So leptin is a signal for you to stop eating, or a satiety hormone. And then ghrelin, produced by the stomach, is a hormone that tells your brain, yes, I'm hungry, I need to eat. So in this section, we'll look at the stomach physiology and regulation. And as part of the regulation, there are 3 phases of gastric control. So to summarize what goes on in the stomach is a holding tank in which food is saturated and mixed with gastric juice, which contains acid and pepsin. And then the food from the stomach is slowly moved into the small intestine. So chemical digestion, as we covered before, is when you're actually breaking chemical bonds and breaking large molecules down into their subunits. So if you remember, when the food is in your mouth, you have salivary amylase, which starts the digestion of carbohydrates, and you have lingual lipase that starts the digestion of lipids. So both of these enzymes can continue working even after you have swallowed the food and it has gone into your stomach until the pH falls below 4.5. And at that point, these 2 enzymes get inactivated. And so depending on how large of a meal you eat, this could be anywhere from 1 to 2 hours after you have eaten a meal. So when you eat food, most of the food you eat is probably going to be close to a neutral pH unless you're eating very acidic foods, things like tomatoes or citric acid or like an orange. But most of the food you eat is going to be fairly neutral pH. So when you swallow that food and it goes to your stomach, it's going to initially raise the pH in your stomach, and then your stomach has to produce that hydrochloric acid slowly, and then the pH drops. So until that pH drops, the salivary amylase and lingual lipase continue their digestion of carbohydrates and lipids. Then once the pH reaches 2, so the pH is dropping because of the production of the hydrochloric acid. Once the pH reaches 2, the amylase and lipase are deactivated, but then the pepsin gets activated. So if you remember, pepsinogen converts into pepsin once the pH gets down around 2, and this begins the preliminary digestion of proteins. Proteins are only partially digested, so they aren't broken all the way down into their individual amino acids. Instead, pepsin breaks them down into smaller polypeptide chains. So in the gastric juice of the stomach, with the acid and the pepsin, we're going to have these really long proteins, which are really long chains of amino acids, and pepsin is going to break them down into these smaller polypeptides that are only a few amino acids each. You also have mechanical digestion that takes place inside the stomach. So the churning and mixing action of the stomach, which is that type of movement called segmentation, is going to help distribute the acid and enzymes throughout the chyme. A lot of people mistakenly think that you actually absorb food in your stomach, and this is incorrect. No nutrients are absorbed by the stomach. So remember that. No nutrients are absorbed by the stomach. So why? Well, the epithelial cells in the stomach are covered by a thick layer of mucus. So remember, they have this mucus carpet, which helps to protect them from the acid and enzymes. So the actual epithelial cells never are really directly exposed to the chyme. There is this layer of mucus between them and the chyme. Secondly, the epithelial cells lack specialized transport mechanisms, so they don't have the carrier proteins and the channels that are found in the cells that are lining the small intestines, which is where nutrient absorption actually takes place. Third, the gastric lining is mostly impermeable to water. And fourth, digestion is not yet complete in the stomach. The carbohydrates, lipids, and proteins at this point are only partially broken down, and they have to be completely broken down into their subunits or monomers before you can have absorption. There are some drugs that can be absorbed in the stomach. Alcohol is one of them. So alcohol is lipid soluble, so it can go directly across the cell membrane of the stomach epithelial cells. And for this reason, if you drink alcohol with a meal, the alcohol is going to be absorbed far sooner than any of the nutrients get absorbed. Aspirin, which is salicylic acid, is also lipid soluble and can be absorbed in the stomach, but it can also alter the mucus layer and promote epithelial damage, so prolonged use of aspirin can lead to peptic ulcers and bleeding. And you should never take aspirin on an empty stomach because aspirin is acid and you're making acid in the stomach anyway. So adding acid on top of acid will just make the pH lower and make you more likely to develop a peptic ulcer. So the production of acid and pepsin by the gastric mucosa is controlled by the central nervous system, also by short reflexes of the enteric nervous system, and hormones released by the digestive tract. So when we look at the regulation of gastric or stomach activity, we can divide it into three phases. The first phase is the cephalic phase, and this phase starts when you see, smell, taste, or even just think about food. So in the cephalic phase, we're going to be involving the central nervous system and commands that it sends to the stomach through the vagus nerve, which is cranial nerve number 10. So the parasympathetic nervous system will activate the mucus cells, which make mucus, the chief cells, which make pepsinogen, the parietal cells, which make the hydrochloric acid, and also the G cells, which make the hormone gastrin. And so all of these stimulations are going to increase the production of gastric juice. So you start producing the enzymes and acid just by seeing, smelling, tasting, or thinking about food before you've ever even swallowed anything. The second phase is the gastric phase, and this begins with the arrival of food into the stomach. The gastric phase can last as long as 3 to 4 hours, depending on how large of a meal you have eaten. And the stimuli to start the gastric phase is going to be distension of the stomach, which is detected by stretch receptors, and also an increase in pH, which is going to be detected by chemoreceptors. So again, remember, when you eat food, what you're eating is probably close to a neutral pH, and the stomach is normally acidic, so a low pH. So when food first comes into the stomach and it hasn't mixed with the acid yet, it is going to raise the pH. Also, the presence of undigested material, especially proteins, can also trigger the gastric phase. So if we look first at the neural response, so with the neural response, the stretch receptors and the chemoreceptors trigger these short reflexes within the enteric nervous system. And again, remember, the enteric nervous system is made up of the submucosal and myenteric plexuses. And this is going to activate the mucus cells, the chief cells, the parietal cells, and the G cells. And as we mentioned before, mucus cells produce mucus, chief cells produce pepsinogen, parietal cells produce hydrochloric acid, G cells produce the hormone gastrin. The neural response will also trigger mixing waves in the muscular layer, so it will cause those muscles to contract and relax so that the stomach starts to churn. And then with the hormone response, it's going to be mediated at the gastric phase by gastrin. So the gastrin is going to further increase the secretions of the chief cells and the parietal cells, and gastrin also stimulates gastric motility by increasing the mixing waves. The next phase is the intestinal phase, and this is triggered by the entry of the chyme into the duodenum, or the first segment of the small intestine. So the function of the intestinal phase, again, remember we're talking about gastric activity regulation. So the function of the intestinal phase is to control the rate of gastric emptying, so the duodenum can efficiently mix the chyme that is coming in with buffers and enzymes. You don't want to dump the entire contents of the stomach into the small intestines all in one go, because that would just be too much acid and it could easily eat away at the walls of the small intestine. So the stomach lets it out a little bit at a time so that the duodenum can mix it with buffers and neutralize the pH, and also mix it in with a bunch more enzymes before moving it along. And then once that little bit has been processed and moved along, then it's ready for another little bit to come out of the stomach. So the small intestine only wants the chyme from the stomach in small amounts at a time. So the function of the intestinal phase is to control that rate of emptying so the duodenum doesn't get overwhelmed. So the stimulus for the start of the intestinal phase is going to be distension of the duodenum, which is going to be sensed by stretch receptors. And then chemoreceptors will sense a lower pH in the duodenum from the stomach acid that has just come in with the chyme. So the neural response is something called the enterogastric reflex, and this is going to inhibit the gastric contractions. So we're going to stop the mixing motions of the stomach, and it's also going to close the pyloric sphincter, which is the sphincter between the stomach and the duodenum, so that only a little bit of that chyme gets through and the stomach holds on to the rest until the small intestines is ready. The hormonal response is going to involve several hormones that are released by the duodenum, and I'm just going to introduce you to them here, and we're going to talk about them in detail when we get to the small intestine. So the presence of lipids and carbohydrates in the duodenum is going to stimulate the release of cholecystokinin, or CCK, and gastric inhibitory peptide, or GIP. A decrease in the pH inside the duodenum from the stomach acid coming in is going to stimulate the release of a hormone called secretin. And again, we are going to talk about these hormones in more detail later on, but suffice it to say for now that what they are doing is they are blocking the secretions by the chief cells, the parietal cells, and they're blocking the muscle action in the stomach. And then if you do have a lot of partially digested proteins that come into the duodenum, that will actually stimulate the duodenum G cells to release more gastrin, which will re-increase the stomach activity. So there are also some reflexes involved in the stomach that are controlled by the central nervous system, and both of these reflexes are triggered by stretching in the stomach wall, so by stretch receptors that is in the wall of the stomach. So the first one is called the gastroenteric reflex, and this increases motility and secretion in the small intestine. So basically think of it this way: you have food that arrives in the stomach, and so the body is getting the small intestine prepared because that is going to be the next stop for that food. So when the stomach's done with it, it is going to go into the small intestine. So we're essentially getting the small intestines ready. And the gastroileal reflex triggers the opening of the ileocecal valve, which is a sphincter that controls movement of materials from the small intestine into the large intestine. And this is why usually after you have eaten a meal, you feel the urge to defecate. And that is because your ileocecal valve has opened with this reflex. Food that you ate at an earlier meal is going from your small intestines to your large intestine. That is pushing the large intestine stuff forward, and then that is going to give you the urge to defecate. The vomiting reflex is also controlled by the central nervous system, the medulla oblongata, and vomiting is also called emesis. So the vomiting reflex is triggered in response to irritation of the fauces, the pharynx, the esophagus, the stomach, or the proximal segment of the small intestine. And while you are engaged in the vomiting reflex, the pyloric sphincter relaxes. So that's the sphincter between the duodenum and the stomach. and then strong reverse peristaltic waves push the contents of the duodenum and the stomach back up into the esophagus and then into the pharynx and then out of the oral cavity. A couple of final thoughts about the stomach before we move on to the small intestine. Meals that are high in protein will stay in the stomach longer because the body will be able to sense that you have a lot of undigested peptides, which will keep the food in the stomach longer so that pepsin has a chance to work on it. So this is why if you eat a meal that is high in protein, you won't be hungry for a while. Whereas if you eat a meal that is like all carbohydrates, so like you just eat rice or noodles or something, you may be hungry in just an hour or two because that passes through the stomach very quickly. And if you drink alcohol or caffeine with a meal, both of these drugs stimulate gastric motility and cause the stomach to empty out earlier, which again can cause you to feel hungrier sooner than you otherwise would have. In this section, we're going to look at the pancreas, and we're looking at the exocrine function of the pancreas. We covered the endocrine function earlier this semester. So the pancreas is an organ belonging to both the digestive and endocrine systems, and so it has both exocrine and endocrine functions. So we're going to focus on the exocrine functions for this chapter, and the exocrine functions are that it produces digestive enzymes, and it also produces buffers to help to neutralize the acid in the chyme that is coming from the stomach. So if we look at the anatomy of the pancreas, the head sits in the duodenal loop, and the reason why we're taking a short detour to talk about the pancreas and liver before we move on to the small intestines is because the pancreas and the liver both secrete substances into the duodenum. The pancreas is one of those organs that is retroperitoneal, meaning that it sits behind the peritoneal membranes. And it has a lumpy lobular texture and a thin transparent capsule of connective tissue. Partitions of this connective tissue also divide the pancreas into distinct lobules, which gives it its little lumpy appearance. So the pancreatic duct delivers the exocrine secretions of the pancreas to the duodenum. It joins up with the bile duct that comes from the liver, and both ducts empty into the duodenal ampulla, which can be seen inside the duodenum as a little swollen area called the duodenal papilla. Some people have an accessory pancreatic duct, but the majority of people just have the one main pancreatic duct. So if we look inside the pancreas, we can see that there are two broad types of cells. The acinar cells are the exocrine cells of the pancreas that produce the digestive enzymes and the buffers, and these account for 99% of all pancreatic cells. So the pancreatic duct, and a branch of it is shown here in this image. The pancreatic duct branches begin in these little pockets called pancreatic acini, and which are lined by these acinar cells. So the acinar cells produce pancreatic juice, which is an alkaline mixture of digestive enzymes, water, and ions. The pancreas also has pancreatic islets that contain the cells that make up the endocrine tissues of the pancreas, but these only account for about 1% of all of the pancreatic cells. And if you look at the histology of the pancreas, you can find the pancreatic islets scattered among the acini. So we covered the hormones that were secreted by the endocrine cells in the pancreatic islets, which included the beta cells and the alpha cells. So the acinar cells secrete about 1 liter per day of pancreatic juice into the duodenum. So if we're keeping track of how many secretions, we're adding another 1 liter of secretion. The release of this pancreatic juice is controlled by duodenal hormones, and 2 of them in particular. So secretin signals the pancreatic secretion of a watery buffer solution that has a pH of 7.5 to 8.8, so it's alkaline. And the purpose of this is to neutralize the stomach acid in the chyme. Cholecystokinin, or CCK, is another duodenal hormone that signals the production and secretion of pancreatic enzymes. So now we're going to look at some of the pancreatic enzymes. One of them is pancreatic alpha amylase, which breaks down starch, which is a carbohydrate. Another one is pancreatic lipase, which breaks down complex lipids. So if you remember, in the oral cavity, we had lingual lipase and salivary amylase, but those enzymes have now been deactivated and broken down by the acid in the stomach. So in order to continue carbohydrate and lipid digestion, we have to secrete some fresh amylase and lipase, and that is done by the pancreas. There are also nucleases that break down RNA or DNA in the food that you eat. And then there are a broad class of enzymes called proteolytic enzymes, which are any enzyme that breaks down proteins. So proteo stands for protein, lytic breaks down. So proteolytic enzymes. These make up 70% of the total enzyme production, and they are always secreted as proenzymes, which is an inactive form of the enzyme, just like we saw with pepsinogen. And proenzymes prevent the proteolytic enzymes from breaking down the proteins inside the cell that is making them, just as we discussed earlier with the pepsinogen. So important proteolytic proenzymes that are secreted by the pancreas include, the first biggie is trypsinogen. Trypsinogen, after it has been secreted into the duodenum, is converted to trypsin, which is a protease. The activated trypsin then activates the other proenzymes. Chymotrypsinogen is converted to chymotrypsin. Procarboxypeptidase is converted to carboxypeptidase. And proelastase is converted into elastase. So notice that proenzymes either have the prefix pro or they end in inogen. And then all of these break down proteins into tripeptides, which are little pieces of 3 amino acids, dipeptides, which are 2 amino acids, and then individual amino acids. So pancreatitis is inflammation of the pancreas. This is an extremely painful condition. And remember, the pancreas is making a lot of proteolytic enzymes. So the damaged exocrine cells leak the pancreatic fluid into the pancreatic tissues, and these proteolytic enzymes start breaking down the surrounding tissue, because remember, cells contain probably more protein than they do anything else, and so these proteolytic enzymes are going to start breaking down the cells and tissues of the pancreas. In 10 to 15% of the cases of pancreatitis, the process can continue unabated and eventually destroy the whole pancreas. And pancreatitis can be caused by blockage of the pancreatic duct. Usually that is caused by gallstones, as we'll talk about a little bit later. Infection, ischemia, and then some drug reactions, and especially alcohol and chronic alcoholism can lead to pancreatitis. So now we'll look at the liver and gallbladder part 1. We'll look at the liver anatomy, liver damage, and the liver blood flow. And I'm breaking this into 2 sections just so a single section doesn't get too long. So let's start by looking at the liver anatomy. So the liver is the largest visceral organ with a large variety of functions. The falciform ligament we mentioned earlier as part of the mesenteries also divides the left and right lobes of the liver. And a portion of the falciform ligament called the round ligament was originally part of the umbilical vein. So if we look at the other 2 lobes of the liver that you can see from the inferior side of the liver, it can help if you look at a color-coded picture like this one. The caudate lobe lies between the inferior vena cava and the left lobe, shown in yellow. So the caudate is shown in blue right there. And then the quadrate lobe lies between the gallbladder and the left lobe, and it has a roughly square shape, which is why it gets the name quadrate, so quad for square. The porta hepatis is also known as the doorway to the liver, and it's this area right here with these vessels going in and out. So this area is going to contain the common hepatic duct, which is carrying bile that is coming out of the liver, and it also contains the hepatic portal vein and the hepatic artery proper that are taking blood into the liver. So remember, we covered this in chapter 21. The hepatic portal vein is not the same thing as the hepatic vein. The hepatic vein is visible up here coming out of the top of the liver. So liver cells are called hepatocytes, and connective tissue divides the liver into about 100,000 liver lobules, and each liver lobule is roughly 6-sided, so it looks like a little hexagon is shown down here. And a liver lobule is the basic functional unit of the liver. And each liver lobule is divided from the others by an interlobular septum, which is shown here in the picture, so you can see it around the 6 sides. So the hepatocytes within the lobule form a series of plates that are only 1 cell thick. And so when you look at the liver lobule as a whole, they look like they're little spokes that are coming out around like a wheel within each lobule. So the exposed surfaces of the hepatocytes do have microvilli for increased surface area. And running between the plates of hepatocytes, as you can see here on the right side, are a bunch of sinusoidal capillaries. So if you remember from earlier in the semester, a sinusoidal capillary has not only fenestrations but also large gaps between the endothelial cells. And sinusoidal capillaries allow large items like the plasma proteins that are made by the liver to move into and out of the bloodstream. So if we take a closer look at a single liver lobule, which is shown up here in this image, so we're zoomed in to one single lobule, you can see that in the sinusoids, and again, these are the sinusoidal capillaries that are running between the single rows of hepatocytes, you have these macrophages called stellate macrophages. They can also be called Kupffer cells. And these are phagocytes that patrol the sinusoids. And so as the blood is flowing through, these phagocytes engulf pathogens, cell debris, and damaged blood cells. And they also store iron, lipids, and heavy metals that are absorbed in the digestive tract. So as mentioned before, the liver lobules are hexagonal, meaning they have 6 sides. And at each of the corners, so there are 6 corners in total, there is a portal triad, which is shown here in the image. And portal triads have one interlobular vein, which is a branch of the hepatic portal vein, one interlobular artery, which is a branch of the hepatic artery proper, and then one interlobular bile duct that we'll talk about in a bit when we talk about bile. So blood comes into the liver from the systemic circuit arteries through the hepatic artery proper, which is going to branch and eventually become these interlobular arteries. And then it's going to run between the hepatocytes within the lobule, and it's going to provide oxygen and nutrients to the hepatocytes, as shown here by this red vessel. Blood is also coming into the liver from the intestines through the hepatic portal vein, and then it's going to branch and become the interlobular vein. And then this is going to flow through those sinusoidal capillaries and allows the blood from the intestines to be filtered by the hepatocytes as it passes through. So the liver is a very essential organ. Any condition that severely damages the liver is a serious threat to life. The liver does have the capacity to regenerate itself, but only in certain circumstances. And there are 2 common liver conditions. One of them is cirrhosis. And in cirrhosis, the liver lobules are replaced by fibrous tissue. So think of it as actually replacing the working hepatocytes with scar tissue. And because you're basically replacing where the hepatocytes would normally be with scar tissue, the liver is unable to regenerate in these conditions, so damage is permanent, and cirrhosis will eventually lead to liver failure. Cirrhosis is most commonly caused by alcoholism, fatty liver disease, and hepatitis. And so the other common liver condition is hepatitis, which is inflammation of the liver, which is most commonly caused by a viral infection. And then untreated hepatitis will lead to cirrhosis. So liver failure is a life-threatening condition. So when the liver gets bad enough that the majority of it is scar tissue and it's no longer functional, this would be considered late-stage liver disease. And at this point, really the only thing you can do is get a liver transplant, and they can hold off the inevitable. There is an option these days to have dialysis that tries to do some of the functions that the liver would normally do, but that is just a short-term fix while you're waiting on a liver transplant. So now let's look at the blood flow through the liver lobules. So as mentioned before, blood is coming into the liver through both the hepatic artery proper and the hepatic portal vein. So the blood is going to pass through these sinusoidal capillaries within the liver lobule, and then it's going to exit out through the central vein in the middle of each lobule. So let's look at the blood flow. So blood coming from the systemic arteries that is full of oxygen and nutrients is going to come in through the hepatic artery proper, which is going to branch and then eventually become these interlobular arteries. At the same time, you've got systemic veins that are leaving the intestines, and you might want to go back and review chapter 21 when we talked about the hepatic portal circulation, if you forgot. But the systemic veins leaving the intestines are all going to come together to make the hepatic portal vein, which is then going to branch into the different interlobular veins. And then the blood is going to go through these sinusoidal capillaries. And then from the capillaries, the normal systemic capillaries and the sinusoidal capillaries, blood is going to drain out the middle through the central vein and then into the hepatic vein. This is where it leaves the liver. And then it's going to drain into the inferior vena cava to return to the heart. So as blood passes through the sinusoids, the hepatocytes are monitoring and filtering the blood, and then they can also make adjustments to the blood and secrete materials such as plasma proteins into the blood. Remember, the liver makes the majority of the plasma proteins. So now we'll look at the liver and gallbladder part 2, and we'll talk about liver functions, production and function of bile, and bile flow, and the gallbladder. So the liver actually has more than 200 individual functions, but don't worry, we're not going to list all 200 of them. Instead, we're going to divide them into 3 broad categories. So the first of these 3 categories is metabolic regulation. So the liver plays an important role in regulating the composition of the circulating blood. So as the blood is passing by the hepatocytes in those liver lobules, they can sense the composition of the blood and make changes. So for example, they can remove and store excess nutrients. They can also correct nutrient deficiencies by mobilizing stored energy reserves or synthesizing new nutrients from scratch. So we can divide the metabolic regulation into a few different categories. So carbohydrate metabolism is aimed at stabilizing blood glucose levels. So the liver is influenced by both insulin and glucagon, as we covered in chapter 18. So for example, insulin tells the liver to take up glucose and convert it to glycogen. Glucagon tells the liver to take the glycogen and break it down into glucose. The liver also has the capacity to make new glucose in a process called gluconeogenesis. The liver also functions in lipid metabolism, so it regulates the levels of triglycerides, fatty acids, and cholesterol in the blood. It also plays a role in amino acid metabolism. So if you have excess amino acids in your bloodstream, the liver will remove them and either break them down or use them to make other compounds. The liver can also function in waste removal. So for example, when it does take the excess amino acids out of the bloodstream, it removes the amino groups in a process called deamination. But in doing that, it produces ammonia, and then the liver converts that ammonia to urea. And then, as we're going to see when we get to the urinary system, one of the functions of the urinary system is to get rid of the urea in the urine. So that's coming up in a future chapter. And so if you think about it, a lot of the components of our urine, the urea, comes from the breakdown of amino groups, which come from amino acids. The liver also functions in vitamin storage. So fat-soluble vitamins like vitamin A, vitamin D, vitamin E, and vitamin K are stored in the liver. So when you take more than you need, the excess is stored. B12 is also stored in the liver. The liver also stores iron, so mineral storage. So when the iron is being recycled from the hemoglobin molecules being broken down, if you have too much iron, the excess is stored in the liver. This is why if you take too much iron, it can damage your liver. And the liver functions in drug inactivation. So drugs that we take, either over-the-counter or prescription medications, are broken down by the liver. And this is why they only last for a certain length of time. So some drugs, it says take every 4 hours. Some might say take every 6 hours. Some might say take every 8 hours. And if you wonder why you've got different time periods for different drugs, that is based on the time it takes for the liver to break it down. So the second big category of liver functions is hematological regulation. So these are functions related to the blood and the blood cells. So the stellate macrophages that are in those sinusoidal capillaries in the liver lobules are antigen-presenting cells that can stimulate an immune response. So the liver does play a role in phagocytosis and antigen presentation, as we covered in chapter 22. The liver also is where the plasma proteins are synthesized, as we covered in chapter 19. So the hepatocytes produce the plasma proteins like the albumins, the globulins, the fibrinogen, the complement proteins, etc., and then secretes them into the blood through those sinusoidal capillaries. The liver also functions to remove circulating hormones. That allows the endocrine response to be turned off. So the liver absorbs and recycles epinephrine, norepinephrine, insulin, thyroid hormones, and steroid hormones, which includes your sex hormones and your corticosteroids. The liver also absorbs vitamin D3, which is called cholecalciferol, and converts it into a form that the kidneys can use to make calcitriol, which is that hormone that is important for blood calcium regulation that we talked about earlier this semester and last semester. The liver also functions to remove antibodies. So remember, we make a lot of antibodies during an infection, but we don't want to keep them around all the time. So when the infection is over with, we do break down those antibodies, and the liver does that. The liver can also remove and store toxins. So the liver takes the toxins out of the blood, and if they're lipid-soluble toxins, they're actually stored in lipid deposits inside the liver. And other toxins, so like water-soluble toxins, are broken down or secreted into the bile. So our last major category of liver functions is bile production, which is the one thing the liver does that is the most closely related to the digestive system. So we're going to spend a little bit more time on bile and what it is used for and why we store it. So the liver synthesizes and secretes bile. A lot of people mistakenly think that the gallbladder is responsible for making bile, but it does not. The gallbladder is just for bile storage. Bile itself is actually made by the liver. So bile is a substance that is composed of water, ions, bilirubin, which comes from the breakdown of the heme groups in hemoglobin, cholesterol, and bile salts. And the part that is the most important for the digestive system are the bile salts. So bile salts are steroid derivatives that are responsible for the emulsification of ingested lipids, so the fat that we eat. Bile salts are synthesized from cholesterol, which is why you end up with some leftover cholesterol in the bile. And emulsification means to physically break down large lipid globules into smaller lipid droplets, which increases the surface area and allows water-soluble enzymes to get close enough to the little pieces of lipids to come in and break the chemical bonds during chemical digestion. So lipid emulsification, you take these large lipid droplets. So remember, oil and water don't want to mix. So think about like when you add oil to water, it forms this large drop on the top, but then the enzymes aren't going to be able to get inside that drop and break the bonds. So we add the bile salts, and the process of emulsification, we separate this large lipid droplet into a bunch of smaller droplets, so now the enzymes can come in and break it down. So the bile salts are reabsorbed once they reach the ileum, which is a section of the small intestine. And then because the blood from the ileum passes through the hepatic portal system, those bile salts are reabsorbed and collected and then recycled by the liver. So the bile salts are about 90% recycled or reused. And enterohepatic circulation is the term used to describe the cycling of the bile salts from the liver to the small intestines and then back to the liver through the hepatic portal circulation. So emulsification is necessary for efficient chemical digestion of fats by lipase enzymes. So lipases are the enzymes that break down fats. So again, if we just had the large lipid droplets, lipase can't get in there to break the bonds. So we mix these large droplets up with the bile salts. That causes lots of tiny little drops. Now the lipase enzymes can get in there, break the chemical bonds, and actually break the fats down into their smallest units, which can be absorbed by the intestinal cells. So as just mentioned before, the bile is produced by the liver, and it's produced by the hepatocytes in the liver. And if we look at this image of a close-up view of one of the liver lobules, again, remember these are all of our little hepatocytes in here that are in these single thick little plates, single cell thick. And they secrete the bile into tiny passages called bile canaliculi, which are these tiny little green passages here. And then the bile caniculi carry the bile to the corners to the interlobular bile duct that is in the portal triad. So notice that the bile flow is opposite to the direction of the blood flow. So blood flow is moving from the portal triad to the central vein. Bile is moving from the hepatocytes outward to the interlobular bile duct. So then all the interlobular bile ducts will converge to form a left and a right hepatic duct. The left and right hepatic ducts will converge to form a common hepatic duct, which leaves the liver at the porta hepatis. The bile can then go in one of two directions. It can either go to the common bile duct, also just called the bile duct, and into the duodenal ampulla for release into the duodenum, or it can go to the cystic duct and into the gallbladder for storage. So now let's take a closer look at the gallbladder. The gallbladder is a hollow pear-shaped organ that stores and concentrates bile prior to its secretion. So the cystic duct is going to be the little duct that carries bile to and from the gallbladder. And then bile is released from the common bile duct, also just called the bile duct, into the duodenal ampulla, which is also where the pancreatic duct is releasing its secretions. So the hepatopancreatic sphincter is a circular muscle that is around the common bile duct, the pancreatic duct, and the duodenal ampulla. So as a reminder from our previous section, we talked about how the pancreatic duct is going to empty into the duodenal ampulla. Well, so is the bile duct. So the common bile duct also empties here, and so the hepatopancreatic sphincter is a circular muscle that can control the release of the secretions from the liver and the pancreas. So the hepatopancreatic sphincter controls release of bile, and pancreatic buffers and enzymes. And then the duodenal papilla is the little small mound that you can see inside the duodenum, where the duodenal ampulla opens up into the duodenum. So now let's review the production of the bile and then its flow, release, and storage. So remember, bile is actually produced by the hepatocytes. The hepatocytes release the bile into those little bile canaliculi that runs between them. The bile canaliculi come together at the portal triad to enter the interlobular bile duct. All the interlobular bile ducts in the liver come together to form the left and right hepatic ducts. The left and right hepatic ducts come together to form the common hepatic duct, which exits the liver through the porta hepatis. From the common hepatic duct, if we want to directly release the bile can then flow into the common bile duct and then out into the duodenum through the duodenal ampulla. However, if we want to store the bile, then the bile will flow up into the cystic duct, which is shown here on this picture, and then into the gallbladder where it can be stored. Now, bile from the gallbladder can also be released, so if we need extra bile, we can reverse the flow and go from the gallbladder out to the cystic duct and then into the common bile duct and then out into the duodenum. So note that if I were to remove the gallbladder, so if I were to have a gallbladder removal surgery, I can still produce bile and I can still release it. I just don't have a place to store it. So it just cannot be stored and released in large quantities, but you can still make bile and release bile even without a gallbladder. But people who have had their gallbladder removed do tend to have difficulties digesting meals that are very high in fat, because if you eat a meal that is extra high in fat, that is usually when you go and release your stored bile so that you have extra bile to break down that fat. And so people without a gallbladder don't have that extra large quantity of bile that they can release when needed. So the release of the bile from the gallbladder is under the control of a duodenal hormone called cholecystokinin, or CCK. This signals the relaxation of the hepatopancreatic sphincter, which causes both bile and pancreatic secretions to enter the duodenum. And the CCK can actually also stimulate contractions of the gallbladder. So CCK levels increase when you eat a meal that is high in fat, so a lot of lipids. So when the hepatopancreatic sphincter is closed, bile is instead going to flow from the liver up into the gallbladder for storage. The gallbladder also absorbs water from the bile, which concentrates the bile that it stores inside of it. And if the bile becomes too concentrated, stones can form, and these are called gallstones. And typically there are two types. There are gallstones that form out of the cholesterol in the bile, and gallstones that form out of the bilirubin in the bile. And so cholecystitis is inflammation that occurs from gallstones that are either so large they're damaging the wall of the gallbladder, or they get stuck and block either the cystic duct or the common bile duct. And in some cases, if the gallstones get all the way down to the duodenal ampulla and they block the duodenal ampulla, that is also going to block secretion of the pancreatic secretions through the pancreatic duct, and all those proteolytic enzymes back up into the pancreas, and you can actually end up having acute pancreatitis. So now we'll do part one of the small intestines, in which we'll talk about the anatomy, the small intestine wall, and segment specialization. So the small intestine is a long muscular tube where chemical digestion is completed and 90% of nutrient absorption occurs. So the small intestine itself produces very few enzymes. So most of the digestive enzymes are produced by the pancreas, and then the liver, which also produces the bile salts that helps the lipase digest lipids. So the small intestine averages 19.7 foot in length, and it fills much of the peritoneal cavity. It is stabilized by the mesentery proper, as we discussed earlier, and it's divided into three segments. The first segment is the duodenum, or the duodenum. The second one is the jejunum, and the third one is the ileum. And we're going to look in detail of these segments in a moment. It takes food approximately 5 hours to pass through the small intestines from the beginning, so from the duodenum to the ileum. So the duodenum is the first section of the small intestine, and it's the one that is the closest to the stomach, and then it's a C shape that wraps around the head of the pancreas. The duodenum you can think of as the mixing bowl. It is very short. It is only about 10 inches in length, and it's going to receive the chyme from the stomach and mix it with enzymes, buffers, and bile that are secreted from the pancreas and liver through the duodenal ampulla. So the primary function of the duodenum is to neutralize the stomach acid and mix in enzymes and bile. The jejunum is the middle part, and it is approximately 8.2 foot in length. This is where you're going to have the majority of the chemical digestion and absorption occur. And so the primary function of the jejunum is chemical digestion and nutrient absorption. And as we're going to look at in a moment, if you look at the lining of the jejunum, you can see that it's got lots of folds, so lots of these circular folds that increase the surface area that is available for absorption. And then the ileum is the last section, which is about 11.5 feet long. Smaller amounts of absorption take place in the ileum. Things like the bile salts and certain types of vitamins and any nutrients that were missed in the jejunum can be absorbed in the ileum, but the primary site of absorption is going to be the jejunum. The ileum ends at the ileocecal valve, which is a sphincter between the small intestine and large intestine. And the ileum has lots of lymphoid tissue in its wall. These are called Peyer's patches, and we talked about these in chapter 22. So the primary function of the ileum is to protect the small intestine from the large intestine bacteria. So let's take a closer look at the wall of the small intestine. It has the same 4 layers that the rest of the digestive tract has, so the mucosa, submucosa, muscular layer, and serosa, but we're going to look at what makes it unique to the small intestine. So first, as mentioned on the previous slide, the small intestine has a lot of these circular folds, which you can see in the picture down here in the bottom right. So these are very abundant in the small intestine lining. They're also called plicae circulares. There are about 800 total folds in the small intestine, so at its densest you can find 2 folds per centimeter. The folds are coated with intestinal villi. So this picture here is not the fold. This is a picture of one of the folds. So within each fold, you've got further folds, which are called the villi. So the intestinal villi are finger-like projections of the mucosa. Then the individual cells that make up the epithelium, which is going to be the outer lining of the mucosa, and this is a simple columnar epithelium. These cells have microvilli on their surface. So I like this picture a little bit better so you can see the different layers of folding. So first we have the circular folds in the wall of the small intestine. So now down here, one of these would be a circular fold, and you can see on the circular fold you've got the villi sticking out. So you can see the villi a little bit better here. Then this would be a single villus. So we have zoomed in to one of the villi. We're looking at a single villus here. And then if we zoom in and look at the individual epithelial cells that make up the single villus, they have microvilli on them. So folds on folds on folds, right? So 3 levels of folds. Big folds, and then the mucosa is folded, and then the plasma membrane of the mucosal cells are folded. So folds on folds on folds. And this increases the total surface area for absorption by a factor of 600. So the total area for absorption in your small intestine is more than 2,200 square feet, which would be this equivalent to the area covered by 10 lanes of a bowling alley. So now let's look at the intestinal villi more closely. And so here's a picture of a single villus. And so if we look at the single villus, we can see that the lamina propria has an extensive network of capillaries. And again, keep in mind that the primary function of the small intestines is going to be absorption of nutrients. And so we need a place for those nutrients to enter the blood supply. So these capillaries ultimately deliver blood to the hepatic portal circulation, which will deliver it to the liver so that it can be filtered by hepatocytes before it joins the general circulation. Each villus also has a single lacteal, which is the green vessel shown in this image. So the lacteal is a central lymphatic vessel that absorbs materials that cannot enter the bloodstream directly. Usually this is going to be fatty acids. So lipids are packaged up with proteins during digestion into small packages called chylomicrons, and these are absorbed into the lacteals. And so lacteals has the same prefix as like lactation and lactose, which are words associated with milk. And so they got this name because when you cut them open and look at the lymph on the inside, it has a whitish color due to the high level of fats. So early anatomists thought it looked like milk, and that's why these vessels got called lacteals. So chylomicrons are very large lipoproteins. So during the digestion stage, triglycerides, cholesterols, and phospholipids all get packaged together with proteins. And then once these enter the lacteals and then eventually enter the bloodstream as a whole, they can be converted into LDL, which is low density lipoproteins, or HDL, which is high density lipoproteins. Also, the small intestinal microvilli, so remember the individual cells of the villus have microvilli, which you can see in this picture. If you look very closely, you can see the little fuzzy border. Those are the microvilli. They have brush border enzymes that are embedded in the plasma membrane of those microvilli that can do the final chemical digestion steps before the actual absorption takes place. And so when you look at a microscope picture shown up here, this is called the brush border, and this is the border of microvilli on the outside of all of the cells. So intestinal glands are located at the base of the villi, right? So these are the individual villi. So at the base is where we have the intestinal glands. These can also be called intestinal crypts. And in this area, you're going to find stem cells that will divide to replace damaged epithelial cells. So just like in the stomach, the epithelial cells in the small intestine don't live long, so they're continuously being shed and replaced. At the bottom of the intestinal glands, you can find Paneth cells. The Paneth cells release defensins and lysozymes, which are proteins that play a role in innate immunity. And in this other picture I have that shows you one of these intestinal glands, you can see that if you zoom into a Paneth cell, you see a lot of these little granules, which are vesicles that are waiting to release these compounds in the case of a bacterial infection. So these compounds can kill certain types of bacteria. You can also find enteroendocrine glands inside the intestinal glands, and these are going to produce intestinal hormones, which we're going to cover in the next section. And then you may also see goblet cells in the epithelial cell layer, and these are cells that produce mucus. So now let's look at the specialization of the different segments within the small intestine. So we're looking at the specialization of the lining. So in the duodenum, you can also find duodenal submucosal glands, which are glands in the submucosa of the duodenum that produce copious amounts of mucus to protect the duodenum from the acid and the chyme that is coming in from the stomach. So in this little histology slide up here, they're calling these Brunner's glands, but this is the same thing that your textbook is calling the duodenal submucosa glands. So this is going to be a gland in the submucosal layer that is producing a lot of mucus. So this mucus also contains bicarbonate ions, which helps to neutralize the pH of the acid coming in from the stomach. So the duodenum has very few circular folds, and the villi are relatively small. So the duodenum, again, is mainly a mixing bowl. It is not specialized for absorption. Then if we look at the jejunum, remember the jejunum is the part where the majority of the chemical digestion and absorption is going to take place. So the first half of the jejunum has very prominent circular folds and villi, so much longer villi. The villi have this finger shape. They're very well developed, and there are no glands in the submucosa. The size of the folds in the villi gradually decrease in size over the second half of the jejunum. And then you get to the ileum. And in the ileum, the distal half completely lacks circular folds, and the circular folds in villi in the proximal half are going to be shorter and less well defined. What stands out in the ileum is that the lamina propria contains these aggregated lymphoid nodules called Peyer's patches, and as you can see in this picture over here of this histology slide, the Peyer's patches are very pronounced. And they are most abundant in the terminal portion right before the ileum joins up with the large intestine. So this will be part 2 of the small intestine, where we'll look at chemical digestion, mechanical digestion and motility, and regulation by hormones. So about 1.8 liters of intestinal juice enters the intestinal lumen each day, and this is primarily going to come from water that enters the intestine by osmosis. So remember, water moves to an area of high solute. So when food enters the intestine lumen, that increases the solute concentration inside the lumen, and so water will actually flow from the interstitial fluids into the lumen. There are also secretions from the intestinal glands. So note that this intestinal juice is on top of the stomach juice, the saliva, the pancreatic juice, and the bile that we have already released. So enzymes secreted by the pancreas into the duodenum do most of the chemical digestion. But as mostly digested molecules come into contact with the brush border, and remember the brush border is those microvilli on the villi cells, there are brush border enzymes that are in the plasma membrane of these epithelial cells that will finish the chemical digestion process. So here's an example. So let's say you eat a large carbohydrate, like let's say you eat something made of starch, which is a bunch of glucose molecules stuck together. Initially, things like amylase are going to break the starch down into maltose, which is a disaccharide made up of 2 glucose molecules. So it's partially digested, but not yet completely digested. And then on the brush border, which is going to be those microvilli on the epithelial cells, you'll have brush border enzymes. And in this example, we're looking at maltase, which does the final breakdown of maltose into individual glucose molecules. And once I have individual molecules, they can be taken into the epithelial cells and then passed into the bloodstream. So the small molecules from the chemical breakdown by the brush border enzymes are then absorbed. One brush border enzyme is called enteropeptidase, and this brush border enzyme is actually required to convert trypsinogen, which was released by the pancreas, into the active proteolytic enzyme trypsin. So again, here's our small intestine epithelial cell with the microvilli. So another brush border enzyme is the enteropeptidase. So when the enteropeptidase sees trypsinogen, it activates it into trypsin. And then as we discussed in the pancreas section, trypsin then activates other proenzymes. Like, for example, trypsin can convert chymotrypsinogen into chymotrypsin. So peristaltic contractions push the intestinal contents forward, and this is primarily controlled by local short reflexes within the enteric nervous system and not the CNS. but the CNS can have an effect. So the parasympathetic nervous system can speed up local peristalsis and segmentation, and the PNS can also prepare the small intestine for activity by increasing enzyme and buffer production in advance of food arrival. So remember, the parasympathetic nervous division is the rest and digest system. The sympathetic nervous system can inhibit small intestine activity. So keep in mind that the parasympathetic nervous system is going to send excitatory signals to non-sphincter muscles within the intestinal tract. So that is going to promote motility, peristalsis, and segmentation. And the sympathetic division will inhibit those non-sphincteric muscles. So it will inhibit segmentation and peristalsis, but it will actually excite the sphincteric muscles to keep them closed. So now let's look at the duodenal hormones that we've already mentioned quite a few times already. So the duodenal hormones are released by enteroendocrine glands in the intestinal glands of the duodenum. There are more than 5 hormones that the duodenum secretes, but we're going to focus on the major 5. The first one is gastrin, which is secreted by G cells in response to large amounts of incompletely digested proteins coming into the duodenum. But remember, gastrin is also produced by G cells of the stomach. So gastrin is produced in both the stomach and the duodenum. So gastrin has the effect of increasing the production of acid from parietal cells and pepsinogen from chief cells in the stomach. It also increases the stomach motility and mixing. So the next duodenal hormone is the gastric inhibitory peptide, or GIP. GIP is secreted when fats and carbohydrates, especially glucose, enter the duodenum. So it has several effects. In the stomach, it inhibits stomach activity, so it stops the motility and acid and enzyme production. In the pancreas, GIP stimulates secretion of insulin from the beta cells in the pancreatic islets, so from the endocrine portion of the pancreas. In the duodenum, GIP stimulates the duodenal glands to increase their production of mucus. GIP also has some other effects. It can signal lipid synthesis in adipose tissue and increased glucose use by skeletal muscles. So kind of think of GIP as inhibiting the stomach, but also getting ready to promote glucose uptake and use. So the next one is secretin. Secretin is secreted when chyme enters the duodenum, and it has a few effects as well. Its primary effect is on the pancreas. Secretin stimulates the pancreas to produce more alkaline buffers in order to neutralize the acid in the chyme. But secretin also promotes production and secretion of bile by the liver, and it decreases motility and the secretion of the gastric glands in the stomach. The next hormone is cholecystokinin, or CCK. This is secreted when chyme enters the duodenum, and especially when it contains lipids and partially digested proteins. So CCK also has several effects. In the pancreas, it accelerates the production and release of all types of digestive enzymes. It increases contractility of the gallbladder, so makes the gallbladder contract to expel bile. It relaxes the hepatopancreatic sphincter so that the pancreatic juice and the bile can enter the duodenum. And then in the stomach, it's going to decrease the motility, which will reduce the rate of the stomach emptying its contents into the small intestine. CCK also appears to have some central nervous system effects in which it can reduce the sensation of hunger. And then the last of the 5 major ones is vasoactive intestinal peptide, or VIP. This one is also secreted when chyme enters the duodenum. And its effects in the small intestines are to increase the secretion of the intestinal glands and to promote capillary dilation, which is going to help with the absorption of nutrients. So think vasoactive means it's acting on the capillaries, which are vessels. So it's promoting capillary dilation. It also can reduce acid production in the stomach. There is a sixth hormone, and I'm just including it here. It's not one of the 5 major ones, but enterocrine stimulates alkaline mucus production by the duodenal submucosa glands when the acidic chyme enters the duodenum. So here's the picture from your textbook showing the major hormones of the duodenum. And then here is a picture to summarize all of the hormone activity. So when food enters the stomach produces gastrin, and don't forget that the duodenum can also produce gastrin, and gastrin is going to promote the acid production by the parietal cells and the pepsinogen production by the chief cells. Gastrin also stimulates gastric motility and mixing waves. And then, so gastrin is really this one pro-stomach hormone. The other 4 hormones are all sort of anti-stomach. So once the chyme enters the duodenum, we get the gastric inhibitory peptide, which inhibits stomach activity, but it also stimulates the release of insulin from the pancreas, which is going to get cells in the body ready to take up glucose. So it facilitates nutrient utilization by the tissues. Secretin and CCK are going to work together to release pancreatic enzymes and buffers, and bile secretion and ejection of bile from the gallbladder. That's going to help with the breakdown of the macromolecules in the food that you eat. And then vasoactive intestinal peptide is going to dilate the intestinal capillaries. So between breaking the food down into its little components and then dilating the capillaries, we are facilitating nutrient absorption. Hang in there, we're getting close to the end. So this is the last stop on our passage through the digestive tract, and we're going to look at the large intestine, part 1, the anatomy, the lining, and then talk a little bit about a colonoscopy and colorectal cancer. So the large intestine begins at the end of the ileum and then ends at the anus. So the overall functions can be summed up by saying that it stores digestive waste and reabsorbs water. The average length of the large intestine is 4.9 feet. It has 3 segments that we're going to look at separately. The first one is the cecum, then the colon, and then the rectum. So the cecum is this first portion here. It's an expanded pouch that is just inferior to the ileocecal valve. Material enters the cecum through the ileocecal valve from the ileum of the small intestine. And the appendix is a collection of lymphoid tissue called MALT that is attached to the posteromedial surface of the cecum. Then the colon is the main part of the large intestine. It has a larger diameter and thinner walls than the small intestine. And the colon, one of its significant features are these little pouches called haustra. So you can see them all along the length of the colon. And these are pouches within the walls that permit the colon to expand and elongate as matter moves through it. The haustra are created by the actions of the tenei coli, which is longitudinal bands of smooth muscle shown here in the picture. So when the muscle contracts, it can cause the formation of these haustra. So the colon can be subdivided into 4 segments, and here's a picture color coding these segments. So we start after we leave the cecum, we go into the ascending colon, in which matter is going to be moving upward, so it's ascending. Then it's going to go through the transverse colon from the right side to the left side. Remember, when looking at pictures like this, right and left are reversed. Then it's going to pass downward through the descending colon. And then finally, it's going to go through the S-shaped sigmoid colon. And then we'll talk about the rectum in a moment. So the last part of the large intestine is the rectum. This is the last 6 inches of the digestive tract. And the rectum is an expandable organ that is responsible for the temporary storage of feces. So the very last portion of the rectum right here is called the anal canal. And there are two sphincters. There is an internal anal sphincter, which is this innermost muscle. This is made of smooth muscle, and so since it's smooth muscle, it is not under voluntary control, so it is an involuntary muscle. And then you have this outer ring of muscle called the external anal sphincter, and this is made up of skeletal muscle, so you do have voluntary control over this one. The anus is the exit from the anal canal. The anal canal has a lot of veins in it, and so as a result, if you have high pressure in these veins, like if you strain a lot during bowel movements, this can lead to hemorrhoids, which we talked about in chapter 21. So now let's look at the actual wall of the large intestine. And again, we have the same layers that we've had before, the mucosa, submucosa, muscular layer, and serosa. Although in the muscular layer, now we have added the Tenei coli, which wasn't present in the other sections. So we have a lot of large lymphoid nodules or malts in the lamina propria of the mucosa. So here's one of these aggregated lymphoid nodules shown right here. There is also no villi or circular folds on the lining of the large intestine, so it looks pretty flat. There is an abundance of goblet cells in the epithelial lining, which produce mucus. And then there are also lots of these little intestinal glands. So these are the glands down here, and then these little dots up here are the openings to the glands, the opening to the epithelial surface from the glands. So there are distinctive intestinal glands that are dominated by even more goblet cells making mucus. And the intestinal glands in the large intestines tend to be deeper than those found in the small intestine. So there are no enzymes produced or used in the large intestine, and all of that mucus production helps to lubricate the passage of the fecal matter, because as it moves through the large intestine, we're actually taking water away from it, so it becomes drier and more compact. And so the mucus production helps to keep it moving along. So colorectal cancer is the third most common cancer in the U.S., and the best defense is early detection and prompt treatment. So if you ever have blood in your stool or blood in your feces, you should always get yourself checked out. And colorectal cancer will develop from what is called a polyp. And a polyp is a projecting growth of tissue that usually arises in a mucous membrane and then is usually found in the colon and rectum of the digestive tract. So here is a picture of several different types of polyps in different stages. And for the most part, polyps are typically benign, but they do have the capability to develop into malignant cancers. So a colonoscopy is a visual examination of the entire colon, where they will go up into the colon with a colonoscope that has a little camera on it. This allows them to visualize the walls of the colon, as well as to go ahead and take biopsies of any polyps that they find. So colonoscopies are useful for screening for colorectal cancer, and it's recommended that everyone get one by the time they turn age 50, and if there's no problems, they suggest you continue to have them every 10 years. And then this is the large intestine part 2, where we will look at the microbiome and vitamins, as well as waste and the defecation reflex. So the large intestine accounts for 10% of the total absorption. Remember, the small intestine does 90% of the absorption. Reabsorption of water is a very important large intestine function. So we have secreted a lot of water along the path of the digestive tract, and the large intestine is where we can make sure that we take back as much as we can. So about 1,500 milliliters of material enters the large intestine every day, but only 200 milliliters of feces leaves. So we are reabsorbing about 1.3 liters of water from the large intestine every day. So the human microbiome is the term that we use to refer to all of the microbes, bacteria, fungi, and viruses that live in and on the human body. Most of the gut bacteria live in the large intestine. They can break down matter that we can't digest, so stuff that passes undigested through the small intestine, and then while they are breaking down that particular undigested matter, they produce vitamins that we can use. So there are 3 important vitamins produced by our large intestine bacteria. Vitamin K is a fat-soluble vitamin necessary for making clotting factors, and intestinal bacteria produce about half of your daily vitamin K requirements. Biotin is a water-soluble vitamin that is important in glucose metabolism. And vitamin B5, which is also called pantothenic acid, is a water-soluble vitamin required to make steroid hormones and some neurotransmitters. The large intestine can also help to reabsorb bile salts that were missed by the ileum. So when the bilirubin that is secreted into the bile, so as a quick review, bilirubin comes from the breakdown of your old and damaged red blood cells, and so when you take the heme off the hemoglobin, it gets turned eventually into bilirubin. The liver filters it out of the blood, puts it into the bile, and then it is secreted into the digestive tract through the bile. And when the bilirubin reaches the large intestines, bacteria convert it into compounds called urobilins and stercobilins, which is what gives your feces a yellow-brown or brown color. Other bacterial waste products give feces its characteristic odor. And one of the biggest things that we cannot digest is fiber. So fiber is indigestible carbohydrates, usually cellulose, which we don't have enzymes for. And cellulose is found in plant cell walls, so that is why you get fiber from eating different types of fruits and vegetables. Fiber provides a good food source for your large intestine bacteria, and the bacteria that feed on the fiber produce flatus, which is also called gas. So when you eat foods that are very high in fiber, like beans, this will result in an increased bacterial gas production and an increase in flatulence. If the gas can't get out and you end up with trap gas, that can lead to distension of the colon, which is called bloating, as well as cramps. So when you swallow air while you are eating and air goes into the esophagus and stomach, that causes belching or burping. And then the bacteria that are breaking down undigested food in your large intestine is the reason why you have flatulence. Also, when food passes through the small intestine too quickly, so when the motility gets really ramped up and the food goes through so fast it can't be fully digested and absorbed, or it's left undigested because you have a lack of enzymes, or maybe you don't have enough bile because you had your gallbladder removed, you have more material that goes into the large intestines for the bacteria to eat. So this gives the bacteria in the colon a larger food source, so they're able to eat and multiply more quickly, and this is why certain conditions can lead to bloating, cramps, and gas. So we talked about this earlier when we were looking at the stomach regulation. The gastroileal reflex moves materials into the cecum from the small intestine every time you eat. And then once the material is in the large intestine, peristaltic waves are pushing it forward, while at the same time you have segmentation that's going on that mixes the contents of the individual haustra, or the little pouches of the colon. And this is called haustral churning. Every time you also eat, you have what is called a mass movement. And this is a powerful peristaltic contraction that moves materials from the transverse colon to the rest of the colon. And this mass movement action is triggered by the distension of the stomach and duodenum. So when you're eating and putting food into your upper GI tract, it is stimulating the lower GI tract to move matter forward. So these mass movements will force feces down into the rectum, and can trigger the defecation reflex. So the defecation reflex is triggered by the distension of the rectum. The urge to defecate will occur when rectal pressure reaches 15 millimeters mercury. It involves both a local short reflex within the enteric nervous system and a CNS reflex that is part of the parasympathetic division. And then in order for defecation to occur, both the internal and external anal sphincters have to relax. And if you remember, I said earlier that the external anal sphincter, you have voluntary control over it, so you can decide when you want to defecate after you feel the urge. However, once the pressure goes up to 55 millimeters mercury, so you first feel the urge at 15, once the pressure reaches 55 millimeters mercury, then it becomes involuntary. You no longer have any control over it, and you will defecate no matter what. And this is actually how defecation occurs in infants who haven't yet learned to control their external anal sphincter, and then adults who have spinal cord injuries and can't control their external anal sphincter. This is how they defecate. So this is the last section, and this is a summary and a conclusion, and we're going to do this by reviewing chemical digestion and then talking about water balance. And so this is an example also of how you can help to organize the material, to pull together material from multiple sections of this very long lecture. So we're going to review the different major macromolecules and their digestion, starting with carbohydrates. So I'm not going to go over everything that is in these images, but when you are studying, this is an idea of how you can break things down. So we walked through the entire digestive system one organ at a time, but then you can also try to make lists like this to see how everything works together. So you can see here that it's showing some of the stuff the stomach does and the hormones that it secretes, enzymes that it secretes, and same thing for the small intestine, etc. So carbohydrates are also called polysaccharides, and they are long chains of individual sugars, which are called monosaccharides. So when we eat, we typically are eating these very large complex polysaccharides, and the digestion begins in the oral cavity with the secretion of salivary amylase in the saliva. So then the carbohydrates move on to the stomach, and the salivary amylase, if you remember, can work a little while after it gets to the stomach until the pH falls below about 4.5, and then the salivary amylase is deactivated. But in the meantime, it is breaking those polysaccharide chains down into disaccharides, which are two single sugars linked together, and trisaccharides, which would be chains of 3. So then we get to the small intestine, and because the salivary amylase is, by now it's been deactivated and probably digested as well, so we have new amylase that comes in from the pancreas, so we have pancreatic alpha amylase. So we're going to continue digesting the carbohydrates down into disaccharides and trisaccharides in the small intestine. And so amylase, as I just said, only breaks carbohydrates down to disaccharides and trisaccharides, but we need them to be monosaccharides before we can absorb them. So this is where those brush border enzymes play a role. So again, now we're in the intestinal mucosa of the small intestine. So remember, we had all these microvilli on the epithelial cells, and in their plasma membranes is where we find these brush border enzymes. So some examples of brush border enzymes that will break the disaccharides and trisaccharides down into monosaccharides are things like lactase, which breaks down lactose, maltase, which breaks down maltose, and sucrase, which breaks down sucrose, which is table sugar. And did you know if your body cannot produce the enzyme called lactase, then you cannot break down lactose, which is a sugar in milk, and then you become lactose intolerant. So the lactose will pass straight through your small intestine undigested. It'll arrive to your large intestine still intact. It'll be a food source for the bacteria in your large intestine, but as they go to town eating it, they will produce a lot of gas, which can cause bloating and cramps and discomfort. So now we have the brush border enzymes breaking the disaccharides and trisaccharides down into monosaccharides. The monosaccharides are then transported into the cell body of the epithelial cells by facilitated diffusion and co-transport. They cross the epithelial cell border, and then on the other side they are transported out by facilitated diffusion, and then they will enter the capillaries that are inside those villi, and then they are going to be transported first to the liver through the hepatic portal circulation, and the liver will make adjustments as necessary before they enter the general circulation. So that is carbohydrate digestion. So now let's take a look at lipids, and we're going to focus primarily on lipids called triglycerides. And triglycerides are made up of components called fatty acids and monoglycerides, or glycerol is an example. So lipid digestion also begins in the oral cavity with lingual lipase that is produced by the tongue. Again, it's going to go into the stomach where it will continue to break down fats until the pH in the stomach falls below 4.5, and then the lingual lipase is going to be deactivated. So after we get into the small intestine, we're going to need some more enzyme, but we also have bile salts that come in from the liver, and the bile salts will help to emulsify the lipids, so break those large lipid drops down into very small droplets. And then we'll also have pancreatic lipase from the pancreas, which will break these bonds that are holding the fatty acids and glycerol together. So lipase breaks down triglycerides into 3 fatty acid tails and monoglycerides like glycerol. So these monoglycerides and fatty acids will still clump together to get away from the watery environment, and they form these little bile salt enclosed packages called micelles, and these are going to diffuse into the epithelial cells of the small intestine. And then they are actually resynthesized back into triglycerides. So they had to be broken down into monoglycerides and fatty acids in order for them to diffuse into the cell. Then once they are into the cell, other enzymes put them back together and remake the triglycerides. The triglycerides are then packed together with cholesterol and proteins into these structures called chylomicrons. And the chylomicrons are too large for diffusion or transport, so they are secreted by exocytosis, where they will then enter the lacteals, which are those lymphatic vessels that you find inside the villi. And then once the chylomicron is in the lymphatic system, and you can review this in chapter 22, from the intestinal region, those lymphatic vessels are going to eventually dump their lymph into the thoracic duct, which will eventually enter circulation through the left subclavian vein. So that is how lipids get into the blood. So now let's look at proteins. And proteins are long chains of amino acids that are linked together called peptides. And protein digestion is the most complicated and time-consuming. So the actual protein digestion gets started in the stomach, where we have pepsin and hydrochloric acid. So the acid denatures or unfolds the protein so that pepsin can get in and break the bonds between the amino acids. So as long as a protein is folded up in its three-dimensional structure, pepsin can't get in to break the bonds. So acid helps to denature or unfold the protein. So then the digestion will continue once the polypeptides move into the small intestine, and there are additional enzymes that are added that come from the pancreas, including trypsin, chymotrypsin, elastase, and carboxypeptidase. All of these plus pepsin are all considered to be proteolytic enzymes, meaning that they break the peptide bonds between amino acids in these peptide chains. Now, the reason why we have so many different types of enzymes to break down proteins is because there are 20 different types of amino acids, and each amino acid has a slightly different structure, and so it takes different enzymes to be able to get in to break different bonds between the amino acids. So we have multiple enzymes because of the way that we have multiple different sequences of amino acids, and some enzymes can only, like for example, cut next to an alanine. So not all proteins may have alanine as an amino acid. So we've got multiple enzymes. So these enzymes are breaking the peptide bonds, and so we are ending up with these very short peptides, and then in some cases amino acids, single amino acids. So carboxypeptidase is one of those enzymes that will only break a bond between the peptide chain and the very last amino acid in the chain. So it always chops off the last amino acid. So it is one of the enzymes that does help to create single amino acids. Several of these other enzymes, though, you're only going to end up with short peptide chains. Sometimes you may end up with a chain of just 2 amino acids, which is called a dipeptide. And so this is where our brush border enzymes come into play. So as part of the brush border, we have enzymes called dipeptidases that will break those chains of 2 amino acids down into single amino acids. So dipeptidase breaks dipeptides into single amino acids. Once we have single amino acids, they enter the epithelial cells by facilitated diffusion and co-transport. They go across the epithelial cell, and on the other side they exit, again using facilitated diffusion and co-transport. Well, they will enter the capillaries that are in the middle of the villus. And the amino acids will also be transported to the liver first through the hepatic portal circulation before they enter the general circulation. Nucleic acids in your food like RNA and DNA are also broken down, for example, by enzymes called nucleases, but we're not going to really cover that because they make up such a very small percentage of the food you eat. The majority of the food you eat is made up of proteins, carbohydrates, and lipids. So the last thing to talk about is the water balance. So we're going to look at the balance between secretion and reabsorption. So keep in mind that water only moves by osmosis. And if you need to, go back to chapter 3 to review the concept of osmosis. But in osmosis, water moves from areas of low solute to areas of high solute. So water enters the digestive tract about 2.2 liters or 2,200 milliliters in our diet, so in the food we eat and what we drink. And then as I've pointed out, as we have gone through this chapter, there are lots of secretions going on, digestive secretions. So a total of 7,200 milliliters is added in secretions. There was 1.5 liters of saliva, 1.5 liters of gastric secretions, then you have liver, the bile is another liter, the pancreatic juice is another liter, another 2 liters secreted in the intestines, and then there are some mucus secretions in the colon that amount to about 200 mils. So at the top of the digestive tract, after you have first eaten something, eating the food, and when it goes into the lumen of the digestive tract, that's going to increase the solute concentration in the lumen, which is going to help water move into the lumen of the digestive tract because water moves toward the high solute. But then as you start absorbing nutrients, you're taking solute out of the lumen and bringing it into the body. So then you start to reabsorb the water. So about 8 liters of water is reabsorbed in the small intestine, and another 1,250 milliliters of water is reabsorbed in the colon. So as the absorption of those nutrients lowers the solute concentration inside the lumen, water can then move back into the body. So when you have diarrhea, right, that is when your digested food passes through the system so fast that you can't absorb the nutrients. And so because you can't absorb the nutrients, the solute concentration inside the digestive tract lumen stays high, so you don't reabsorb the water either, and you end up losing a lot of water when you have diarrhea. So when everything is working as it should, only about 150 milliliters of water is lost in your feces each day. And that is the end of chapter 24. So again, try different ways of organizing the material to help you with studying it. I know this chapter is a lot of material. It's just the nature of A and P.